Hello, everybody. Welcome back to day 5. I guess you heard that cough. Still under the weather a little bit, but not nearly as bad as last night or the day before. I had chills. I had upper respiratory something or other, and it's almost like flu-like symptoms. But you know what got over it? How I got over that part? Because I'm here in front of you. The excitement for me to be in front of you helped me overcome that lousy virus that's infecting me or whatever. So I'm feeling better today. I still got a little bit ways going, but in any case, I'm fired up for tonight because there's a lot of we're going to finish up genetics or get close to finishing up genetics tonight in these 4 hours. And now, before we proceed here in chapter 2, I do want to say something about Facebook. Again, I've been posting on average 2 or 3 questions a night. And again, only about half a dozen to 10 students respond or put in an option and explain it, which is great. But I'd like to see more participation. There's a lot of people are signed up for that Facebook. I don't understand why people are not participating. It's there. It's for your benefit. And one of the beauties of typing in a response is that when you put in an option, if you give a mistake, then hopefully one of your classmates will say, well, no, I think it's this way. And so you start learning it better. If you start to defend your answer or have to explain your answer, you learn it a lot better. And classmates can help you out. Now, you saw today that one in that Facebook, that one question that dealt with thyroglobulin. And thyroglobulin, when it precipitates in the ER, then because of a mutation, thyroglobulin will precipitate in the ER. And then Syed asked, great question, why wasn't it being degraded by the proteasome? That's a great question, Syed. You're thinking. Why wasn't it being degraded by the proteasome? And then Marsalo, if I pronounced your name incorrectly, I apologize, but she wonderfully went in there and said the issue is location, because the proteasome is in the cytoplasm and the thyroglobulin, which misfolds and precipitates in the ER, it's in a different location. They never see each other. There's this thing called the ER membrane, and the proteasome cannot go through it to degrade the thyroglobulin. So it was a matter of thinking, wow, this thing is precipitating, and as Syed says, why isn't it being degraded? And then you start thinking, well, where is the proteasome at? And that's how you have to think. You can't memorize that kind of information. And it's just a matter of, and this is what the benefit of the Facebook is. We could do a few questions live online. We'll do a few questions tonight. And in fact, at the end of this chapter, we'll do a couple questions. We'll do a couple more questions later on. But we just can't keep doing questions because we have to get to the heart of the information. And then you apply the information after you master it to solving questions and things like that. So again, use the Facebook. It's to your benefit. And it's all free. Questions are there. I go in and give you the answers, and I look at everybody's responses and seeing, is it ultimately we getting to the right answer and so on. Give you a lot of time to look it over and so on. So it's to your benefit. Okay, all right. So now with that in mind, now in chapter 1 in genetics was a very high yield chapter because it dealt with all those modes of inheritance. It dealt with all those terminologies, and you need to know, as I mentioned yesterday, what diseases are associated with those particular terms, like pleiotrophy, locus, heterogeneity. Okay, so it's a high yield chapter, all the stuff of imprinting, major. You could predict, I don't have to be here to tell you, you would predict you're going to get at least one or more questions going to come from chapter 1, right? At least one. So be ready for it. Okay, now chapter 2 deals with population genetics. Now this particular chapter can be, as I mentioned yesterday at the very end, don't read ahead here because you can get bogged down. There's a lot of stuff that's not important in population genetics. But there is one part of it that is extremely high yield. And I'll cover it, don't worry. But the idea is, as long as you're paying attention, I'll tell you why you need to know it and how you might see it on board exams. That has to do with this thing called the Hardy-Weinberg. Now, a lot of students, I know a lot of medical students hate mathematics. Anything that has to do with calculations and equations and things like that, they get turned off. You know, a person just shuts down when they see an equation or something like that. But you're making excuses. Don't shut down here. I'm going to take it a little slow. I can't take it too slow because I got to move on, but I'll take it a little slow. I'll try to be as clear as I can, explaining what's happening here and what does the Hardy-Weinberg do, okay, in this population genetics here. Okay, so before we get to the Hardy-Weinberg equation, there are two terms I need to define. And one term is the term called genotype frequency. And then after that, we'll talk about allele frequency. Now, if I were to, without looking at this paragraph here, if I ask you to explain what does that mean, genotype frequency, you would say, you would think because of the words, it must be the frequency or the percentage of a particular genotype in a population, right? The frequency of a particular genotype, whether it's homozygous normal, heterozygous, or homozygous mutant, a frequency of a particular genotype in a given population. And here it is. I mean, another way of saying it is right here. The genotype frequency measures the proportion, that's percentage, of each genotype in a population. Now, what do I mean? Again, to try to explain this a little bit easier, another way of doing the same thing. As long as you're paying attention, don't get distracted now. Don't go looking at the NFL draft, that kind of stuff. Pay attention. So the idea is, suppose we have two chromosomes that you have, I have, so whatever chromosome number it is, not important. This could be chromosome 5. I got 2 of them. You have 2 chromosome 5s. Your neighbor has 2 chromosome 5s. Now suppose in that particular chromosome, there is a locus of a gene. There's somewhere in it, there's a location of a particular gene. And that's going to be shown in the orange. So that's some gene that's at that particular locus of that particular chromosome. Now the sequence, the DNA sequence of that gene, suppose it's a normal sequence, which most people in the world have. And whatever the sequence is, might be the sequence for insulin, might be the sequence for glucagon, or whatever it is. It's a sequence of a gene, which most people in the world have. But that gene can be mutated. So the genotype at that particular locus, it could be, I'm going to show it here as 1, 2. It could be 1, 1, or it could be 2, 2. What I mean by this is, suppose the genotype, suppose the allele 1, version 1, the DNA sequence of version 1 is the normal sequence of that particular gene. And version 2 is the mutated sequence of that particular gene due to a missense mutation or whatever, deletion or something like that. So at this particular locus, we can have 3 different versions of the genotype. Somebody can have a 1, 1 genotype, and most people would have a 1, 1 genotype in a given population. Other people in a given population might be heterozygous, 1, 2. And other people in this world, much less frequent, they may be homozygous for that particular mutation, and a genotype would be 2, 2. All right, so that's what. That's the issue of the genotype. Okay, now the idea is, suppose there are 100 people. Now, in listening to me right now, live online, there's over 100 people listening to me, and suppose there was just let's say there was 100 people, including me. We're all together. We're listening here, and if I were to take a biopsy of all 100 people, and then put them in 100 separate test tubes, and then measured the genotype in each of the test tubes, how many of the 100 individuals have the 1, 2 genotype, 1, 1 genotype, and 2, 2 genotype? We could do that. We could do that measurement. So, but the idea is, how many of the 100 people, how many had the 1, 1 genotype? And suppose we found that there were 40. So how many people had the 1, 1 genotype? Suppose theoretically, we found that 49 people out of the 100 had the 1-1 genotype. So what's the percentage? What percent of that population have the normal genotype 1-1? Well, you would say 49 divided by 100 is 0.49. Now, if you don't like decimals very well, make it into percentage. 0.449 is the same thing as 49%. 49% of that population of 100 people have the 1-1 genotype. And then in that same population, if we ask the question, how many people are heterozygous for that particular gene? How many have the 1-2 genotype? And suppose we measured there were 42 people that are heterozygous for that particular gene. They have the 1-2 genotype. And so because it's 42 and we measured in 100 people, so 42 divided by 100 is 0.49. Now, if you don't like decimal points, you like percentages better, which I do personally, 42% of the population has the heterozygous phenotype. And then heterozygous genotype. And then lastly, suppose that we look at that 100 individuals. If 49 had the 1-1 genotype and 42 had the 1-2 genotype, the genotype that could be there is the 2-2 genotype. So there has to be the remaining 9 people, and so that would be found. There are 9 people have the 2-2 genotype, and so 9 divided by 100 is 0.09. That means a much smaller 9% of the population of the people listening to me tonight have the homozygous mutated gene. So the idea is in a genotype frequency, the 1-1 genotype by convention is considered that the by the way, so all the numbers should add up to 100%. So in terms of decimal points, it should add up to 1.00. But if you like percentages better, it should add up to 100%, meaning you have accounted for all the genotypes in that population of 100 students listening to me tonight. Okay, so again, the 1-1 genotype is by convention is characterized as the homozygous normal. The 1-2 genotype is the heterozygous carrier status, and the 2-2 genotype is considered the homozygous mutant situation. So that's genotype frequency. It's not hard, just a matter of thinking, well, what does it mean, and this kind of stuff. Okay, now there's a related term called allele frequency. Now what that means is, now this one's a little bit harder to understand what's going on there, but just think about it. It'll start to make sense here. The idea of allele frequency, what does it do? It measures the proportion of chromosomes that contain a specific allele. In other words, of the 100 people that in this room listening to me tonight, of the 100 people in this room listening to me, in that particular chromosomes that have a version 1 or version 2, what percent of the chromosomes that have either version 1 or version 2, what percent of them have version 1, and what percent of them have version 2? Okay, that's allele frequency. So again, so we know there's 100 people in the room, and the genotype frequency we just went through. Now, if you're asked what does allele frequency means, it says of the total chromosomes, of the total chromosomes that have either version 1 or version 2, what percent of them have version 1 and what percent of them have version 2, and should add up to 100%. Okay, so the idea is, let me ask you this. Of the 100 people, continuing on with the 100 people listening to me tonight, of the 100 people that have either version 1 or version 2, let me ask you this. What are the total number of chromosomes? What's the total number of chromosomes that have either version 1 and version 2 together? What's the total number of chromosomes that have either version 1 or version 2? Let me see your answers here. Chromosome number it is, they have the 1-1 genotype, 1-2, or 2-2 genotype. What percent of the chromosomes have, what's the total number of chromosomes? The total number of chromosomes have either version 1 or version 2. Let me see. Anybody? What's the total number of chromosomes? that have version 1 or version 2. Remember, we're diploid, right? Remember, we're diploid, and because we have 2 chromosomes that have either version 1, version 2, that means the total number of chromosomes that have either version 1 or version 2 got to be 200. There's 200 chromosomes. There's 100 individuals, and the total number of chromosomes that have either version 1 or version 2 will be 200. It's not hard. It's not hard. If I ask you how many chromosomes have either a normal gene or a mutated gene, you got to add it. You got to say, well, we're diploid. So how many people are we talking about? And whatever number that number of people is, you multiply by 2 because we're diploid. We have 2 chromosomes that have that particular gene. Okay, it's not hard. Okay, so the denominator will be 200. We'll have either version 1 or version 2. All right, here comes now. Suppose we wanted to know how many of those chromosomes, of the 200 chromosomes that have either version 1 or version 2. What's the number? What's the percentage of chromosomes have version 1 only? Okay, one more time. So if I were to, if the total number of chromosomes is 200, what percent of them have version 1? And so the way to do this is there are 49 people, and they have the 1-1 genotype. And so if you ask in these 49 people, how many of their chromosomes have version 1? Well, because there's 49 chromosomes, 49 people, and the chromosome is present twice with version 1. Therefore, the numerator got to have 2 times 49. There are 2 chromosomes that have version 1 in each of the 49 people. So it's 2 times 49 are the number of chromosomes in those individuals that have version 1. And then we look at the people who and you would say, well, if 70% of the chromosomes have version 1, that means 30% of the chromosomes must have version 2. And that's true. Matter of fact, there's the mathematics of that right there. It's 0.3, which is 30%. Here it is. So there are 9 people that are homozygous mutant. So both of the chromosomes have version 2. So what's the total number of chromosomes in these 9 people that have version 2? And it's 2 times 9, that's 18 chromosomes. And in these people here who are heterozygous, one of the 2 chromosomes is a mutated gene. So 42 times 1, there it is, 42. And divided by total chromosomes, 200, and it comes out to 0.3. So what that means is 0.7, that's the normal, plus 0.3, that's the mutated, equals 1. Now if you like percentages better, that's 70% of the chromosomes have version 1, 30% of the chromosomes have version 2, for total chromosomes of 100. We have accounted for the total number of chromosomes. It must add up to 100%. Otherwise, there's something fishy going on, which they'll never do on board exams. So the idea is that total number of chromosomes should add up to 100%, the percentages here. Okay? So the idea is, again, I know students hate mathematics. I know the students hate mathematics here, but it's not difficult mathematics. It's a matter of thinking here. Okay, so now this is going to be called the allele frequency. The normal gene, the mutant gene should equal to 1. Now we get to the all-important Hardy-Weinberg. What the Hardy-Weinberg is all about is it measures, it predicts the genotype frequency and allele frequency in a non-evolving population. Genotype and allele frequencies in a non-evolving population. Now remember from the previous page, you saw the allele frequency. Remember that? That was P plus Q equals 1. Now here, see these letters P plus Q? By convention, the P is the frequency of allele 1, which is typically the normal allele, and Q by convention is the frequency of the mutant allele, the disease-producing allele. Now you saw that on the previous page. 70% of the population had the normal gene, and 30% of the population have the mutant gene. That's the allele frequency. But now here's like a little equation, P plus Q. Okay, but now comes the Hardy-Weinberg. Now don't get bogged down, but you need to know it. I'm telling you right now, I hear about it on boards frequently, the Hardy-Weinberg. And it's not difficult as long as you understand what it's telling you. Here is the Hardy-Weinberg equilibrium. It's P squared plus 2PQ plus Q squared equals 1. That is the genotype frequency issues. So now what this is about is P squared is the frequency of the genotype 1,1, which is again, that's the genotype frequency. And P squared is the frequency of genotype 1,1, which is the normal genotype. Both genes are normal. 2PQ is the frequency of genotype 1,2. That's the carrier. That's the heterozygous situation. And Q squared is the frequency of the homozygous mutant situation, and that's the 2,2. Homozygous mutant situation is Q squared. Okay, so again, this is genotype frequency here. You saw that on the previous page as well. Remember that issue that there were 49% of the population have the normal gene. There was, in terms of the carriers, there were 42% of the population, and 9% of the population had the homozygous mutant situation. But it adds up to equal to 1. Okay, so everybody with me so far? Is everybody with me? I'm going to pause just to make sure. So far, so good? So far, so good? Don't go ask me to go through the same half hour again or 20 minutes. But if there's some quick question I can answer, ask. But if I can't keep explaining over and over. But if there's a quick question you want me to respond to, I'd be happy to do that. Okay, or use the Q&A pod, and Dr. Donald and Dr. Prashant will be happy to respond in a more lengthy situation. All right, okay, so no questions. All right, now here comes the use of it. What we did was establish what is the Hardy-Weinberg, and now the use of it. Now let me stress here, remember earlier when we talked yesterday, when we talked about incomplete penetrance. If on board exams they ask you a question about incomplete penetrance, you automatically think they're talking about an autosomal dominant disease. Again, technically, it doesn't have to be autosomal dominant. You can read about that and you'd be wasting time. But for your purpose for the boards, when they give you incomplete penetrance, you think autosomal dominant disease is what they're asking me about, like retinoblastoma, that type of stuff. But if they're giving you a question about Hardy-Weinberg, you automatically think it's an autosomal recessive disease that they're asking me about. Autosomal recessive disease. It doesn't have to be. You could use Hardy-Weinberg for X-linked diseases as well, but don't worry about that. They're not going to complicate the situation because they know the Hardy-Weinberg. Most students are turned off about it. They don't like it. But I'm telling you, I'm going to make it simple so you will not fear the Hardy-Weinberg. So the idea is, the Hardy-Weinberg is used for autosomal recessive diseases for your purpose for the boards. All right, now here comes a little number of paragraphs that are in your book. Now it says in the upper vignette, it says a 20-year-old college female college student is taking a course in genetics. She is aware that she has an autosomal recessive disease that has required her to have a lifelong adherence to a diet low in natural proteins, was supplemented with tyrosine and restricted amounts of phenylalanine. Let me ask you this. Based on those two sentences, what disease, everybody should get this one, what disease does she have without looking at the rest of the paragraphs here? What disease did she have? What disease? Oh, this thing wasn't working. Okay, no, I said, that public chat scroll was not working. Okay, good. Now I see. Okay, oh, there you go. Now I see it. When I'm scrolling down, I see everybody say yes. Oh, wonderful. I was wondering, nobody was responding. I thought, man, am I boring them stiff right now? They're falling asleep on me. Okay, yeah, that's right. It is. Well, a little worried, man. All right. So the idea is, that's right. So this woman has She's homozygous for PKU. Okay, so it's autosomal recessive. So both of her genes have are defective for an enzyme. Let me ask you this question. Without looking in the book, what's the name of the enzyme that's defective in PKU? If you forgot, don't worry. We'll get to that later on in metabolism. But what's the name of the enzyme? Everybody should be able to get this one. You don't have to be uh, uh, thinking too hard on this one. That's right. Everybody should be saying phenylalanine hydroxylase. And so when that enzyme is defective, then you cannot make tyrosine. So therefore, tyrosine has to be supplemented in the diet. Okay, now this question has nothing to do with the metabolism of phenylalanine. What she's asking is, she wants to know, okay, now that she know, well, she has PKU, homozygous for PKU. She asks her professor, what are the chances that she would marry a man with a disease-producing allele? Right? I mean, I mean, if I was homozygous for PKU, I would want to know. I mean, what's the probability that I'm going to marry a woman in the general population who's a heterozygous? Because that's obviously going to affect our children, right? So that's the idea. So she wants to know what's the probability that she marries a man with a disease-producing allele. In other words, she wants to know the probability that she marries a carrier, a man with the disease-producing allele. One of his two genes is mutated. She wants to know that. Okay. All right. So it turns out that geneticist says the prevalence, the incidence of PKU in the population is 1 in 1,000. So that is what they're giving her. They're telling her the prevalence of PKU, homozygous PKU, is 1 in 10,000. So if you ask what's the probability of her marrying somebody who's homozygous, it's 1 in 10,000, which is chances are very, very low, obviously. But then what's the chances of her marrying somebody who's a carrier? I mean, that's a logical question. I would want to know it. You would want to know it, right? What's the probability that she marries a carrier? Okay. So now here comes the Hardy-Weinberg. Okay, here's the beauty of the Hardy-Weinberg. So remember, P squared is the homozygous normal situation. 2PQ is the carrier status, which is what you want to know in this question. And Q squared is a homozygous mutant situation. Okay, so again, nothing new. We talked about the Hardy-Weinberg on the previous page. So the hard part about Hardy-Weinberg is what are they giving you in the question and what are you trying to figure out? And what they're giving you Q squared. They're saying Q squared is equal to 1 in 10,000, right? Q squared is equal to 1 in 10,000. And what are you trying to figure out? You're trying to figure out 2PQ, the carrier status. And to figure out the carrier status, we have to figure out what P is, and we have to figure out what Q is. Once we know what P is, the frequency of the normal allele, and once we figure out what Q is, the frequency of the mutant allele, then we just do 2PQ and we get the answer. Now to figure out what Q is, all we got to do, because it says Q squared is equal to 1 in 10,000, all you got to be able to do is square root. Now on board exams, if they ask you to do a square root, it'll be very simple. They know you don't have a calculator, so don't worry about that. So they'll make the mathematics simple. As long as you can do square roots, you'll get the Q here. So the square root of 1 in 10,000 is 1 in 100. That is the allele frequency. That's the frequency of the mutant gene in the general population. Okay. All right. So now we know what Q is. So now we got to figure out what P is. And then we plug the P in, we plug the Q in, and then multiply by 2, we get the answer. All right. Now to figure out what P is, remember that allele frequency equation. P plus Q equals 1. And now we know what Q is. So now that we know what Q is, we can then solve for P. So Q is 1 in 100. That's 0.01. There it is. P plus Q, 0.01, 1 in 100 equals 1. So let me ask you this question here. So what is solving? Again, I don't want to insult your intelligence here, but just to make sure. So solving for P, what's the answer to P? What is the value of P? What is the value of P? Let me see your answer. That's right. That's right. The value of P is 1. Technically, it's 0.99, but for your purpose, just say 1. Again, those are just words. What that means is the frequency of the normal gene for phenylalanine hydroxylase in the general population is almost 1. So again, it's almost 1. Okay. Now let me stress here, on board exams, always say P is 1. Now, if you're now it doesn't have to be, and if you are a geneticist, a PhD in genetics, and you're in genetic counseling, P may not be 1 or whatever. Don't worry about that. Why would they want to make it more complicated? So don't worry about all the complications. Just say for your purpose, P is 1. So what I'm doing is I'm making life simple. P is 1. All right. So here it comes. All right. So 2 times 1, that's P, times 1 in 100 is what? Here, 2 times 1 times 1 in 100, so it's 2 divided by 100 is what? 2 divided by 100 is 1 in 50. There it is. That's the answer. That's the carrier frequency, 1 in 50. Okay. All right. Now, because P is the P is the frequency of the normal gene. Now here comes the question here. Now think, and if you're trying to memorize here, forget about it. But the bigger picture, understand the bigger picture. What this says is, in the people listening to me right now, if there's 100 people in this room, if there's 100 people in this room, what that 1 in 50 says is, there's 1 in 50 probability that people listening to me right now are carriers for PKU. Of the 100 people listening to me right now, 2 of us are carriers for PKU. It might be me and it might be you, and I'll never know it unless I do genetic testing or I mate with somebody who's also a carrier or something like that. Some of my genes have mutations in them, and I may not know it if they're autosomal recessive modes of inheritance. But I have mutations in my gene, and so do you. But the frequency, I mean, just think of this, what this means. It says, of the 100 people right now, listen to me, 2 of us are carriers for PKU. It's staggering if you think about that. It's staggering. Most of us will be normal. Most of us will have the 1 gene type, the normal genes for phenylalanine hydroxylase. But there's going to be 1 in 50 of the people listening to me right now, probability of being a carrier, and 1 in 10,000 people will be homozygous for the mutant gene. That's what the numbers are telling you. Again, if you're trying to just go through the math, go through the equations and so on without thinking, what are you trying to do here? What does it mean? What's the significance? What's the bigger picture? You're just going through the motions. Try to understand what it's telling you here. All right. Okay. So again, it's amazing how this is. 1 in 50 people in the world, 1 in 50 people walking the street. Next time you walk around, if you're living in Chicago right now and you walk down the Miracle Mile, you walk down and just count the people. If you get 2,000 people, there's going to be 4 people there. No, 2,000 people. There'll be 40 people there are carriers for PKU. That's what it means. That's what it means. Pretty significant, right? Pretty amazing. All right, now she knows that the probability of marrying a carrier is 1 in 50. Then she asks another question, which is a logical question that you would ask and I would ask if I was a homozygous. She asks, well, okay, if I marry a carrier, then what's the probability that I'm going to have an offspring who is homozygous with the disease? We have a probability. What is the probability that I will have a child with PKU, homozygous for this disease? All right, so here it comes. So what's the probability that she marries a man who's a carrier? And the answer is 1 in 50. Now then the question becomes, because she's marrying a man, what's the probability that his sperm have a mutant gene for PKU? Remember, Sperm and eggs are haploid. We only have one chromosome, or whatever chromosome number that happens to be. So in that one chromosome, we have one gene. So the idea is of the sperm, what's the probability in this man that she's going to marry? What's the probability that his sperm will be mutated sperm for PKU? And the answer is 1 and 2. One out of every 2 sperm that this man has will have a defective gene. He have a 1 and 2 possibility of having a mutant sperm for PKU, and 1 and 2 possibility of having a normal sperm for phenylalanine hydroxylase. So then you add it up, and it's 1 in 50 times 1.5. The probability that she will have a child with PKU is 1 in 100. That's the probability that she will have a child with PKU. All right, now let me ask you this. So again, the idea is the probability of marrying a carrier is 1 in 50. But now she's asking another question. What happens about having a child with PKU? Then you got to take in consideration the guy's sperm and 1 in 2 possibility of having the mutant gene. Now let me ask you this. What about her eggs? Why are we not in this equation here? Why are we not considering her eggs here? We should, but we're not. Why not? Let me see your hands. You have to think about this, and you'll get the answer. Why aren't we considering her eggs in terms of having a child with PKU? Mithra, not that she's normal. No, she's homozygous with the disease. That's right. It's all of her eggs. All of her eggs are mutated for PKU. She's homozygous. Both genes that she has for phenylalanine hydroxylase are mutated. So technically, it's 1 in 50. That's the probability that she marries a carrier, and that carrier that she mates with, it's a 1 in 2 possibility of the sperm being mutated times 1. Because every egg that she has will be mutated. Okay, so that's why you don't see it times 1, but technically it should be shown, but it's not. But there it is. But again, because all of her eggs will have the mutant gene. All right, now so again, one more time. Now we're going to practice this coming up here. So the idea here is on the Hardy-Weinberg, you're going to see it again. In fact, you're going to see it 3 more times. There's 3 questions coming up here, and so on the hardest part of the Hardy-Weinbergers, don't worry about p squared. Don't worry about p squared, because what does p squared mean? That's the probability of the normal genotype, the normal genes, a pair of genes that are normal. It's going to be it's almost going to be almost 1. Okay, and so on board exams, what they'll do is on board exams, they'll either give you Q squared, that's the prevalence of the disease, and you have to calculate the carrier frequency, or vice versa. They'll give you the carrier frequency, and they ask you what's the incidence of the disease in that population. Okay, so again, on the board exams of the Hardy-Weinberg, it's not hard. It's not hard. And I'm telling you right now, if you try it on board exams, just try it out, and trust me that I gave you the information you need to know. If you get it right, you have such an advantage, because most students won't try Hardy-Weinberg because they get intimidated by the mathematics. It's not hard. All right, so one more time. On board exams, they'll give you either this, and you have to calculate carrier status. They give you the carrier status, and you have to calculate the incidence of the disease. All right, now the first one is a question just to work out on your own. Just to work it out on your own. No selection pod yet. So here's another application of the Hardy-Weinberg. Okay, and what we're going to talk about here is Tay-Sachs disease. I keep telling you, Tay-Sachs disease is often on board exams for many, many reasons. The inclusion body diseases, splicing defect, insertion disease. And here's another reason, because it is often used to illustrate the principle of the Hardy-Weinberg. Now, in a given population of the Ashkenazi Jews, that's a religious group primarily in Eastern Europe. You've been hearing about Ukraine. A lot of people in that country are Ashkenazi, and as a result, and it turns out that 1 in 30, 1 in 30 Ashkenazi Jews is a carrier. Okay, and here comes the question for you. What is the probability? What is the prevalence, I should say? What's the prevalence of Tay-Sachs disease in that population? Not the worldwide population, in that population. Now I'm going to give you 30 seconds to work this out. I'm going to pause for a second, for about 30 seconds or so. Just try it out. Just try it out on your own. Don't be intimidated. Try it. All right. Anybody else? Okay. I see three answers so far that I see that are the correct ones. All right, and here it comes. So again, here comes the Hardy-Weinberg. You got to know the Hardy-Weinberg, okay? So don't worry about p squared. Don't worry about it. So what are they giving you, and what are you trying to figure out? And what they are giving you is they're giving you the carrier status. So just set it up. They're saying 2pq, that's the carrier status, is equal to 1 in 30, right? And they're not hard setting that part up. So 2pq is equal to 1 in 30. All right, so you know that what p is. You know already that p is about equal to 1. So now it's 2 times 1, because p is 1, times q is equal to 1 in 30. Okay. All right, so what are you trying to figure out? So that's the carrier frequency, and all you're going to try to do is figure out the incidence of the disease. That's q squared. So you have to figure out what q is. And there it is. So 2 times 1 times q is equal to 1 in 30. So as long as you can do a little bit of math, q, then solve for the q. The 2 goes to the denominator. So therefore q is equal to 1 in 60. And then all you got to be able to do is just do the square of that, multiplied by itself, and the answer is calculate q squared, and the answer is 1 in 3600. There it is, 1 in 3600. It's not hard. What's hard about it? What is hard about this? Well, I mean, tell me, what is hard about this question? Honestly, it looks formidable. Wow. It's a simple question. It's not hard. As long as you can multiply two numbers with each other, it's not hard, okay? Not hard. Mithra, how did you get 1? Because that's the frequency of the normal gene. Most of the genes for hexosaminidase A in the population is almost 1. Almost 100% of the genes in the population have the normal gene for hexosaminidase A. It's almost 1. Okay, so that's why you say it's 1. It's about equal to 1. Again, what does the P mean? It's the frequency of the normal allele. It's almost 1. Okay. All right. Notice it's not. So for your purpose for the boards, P is about equal to 1. Okay. Don't make it hard. I'm Natalie. Natalie had some good advice the other day. Don't overcomplicate things. This is very easy. Try it out. Okay. Now what we're going to do is we're done. Now, by the way, we're done with the chapter. The rest of the chapter, forget about it. The rest of the chapter has to do with, well, what happens if it's an X-linked dominant or this or the X-linked recessive or what? Forget about it. They're not going to ask that kind of stuff. If you're a PhD in genetics, yeah, they would ask that. But for the boards, why make it harder? Most students won't even do this little thing over here in front of me. So the idea is the rest of the chapter, forget about it. It's the Hardy-Weinberg and its use. Now, what we're going to do is 2 simple questions that come from the end of the chapter. 2 simple questions. And again, don't look at the answer at the end of the book because you're not fooling anybody, but try it out. And each question, I'm going to give you 78 seconds because 78 seconds is the time they give you per question. So I'm giving you the full length time. Earlier, I gave you 60 seconds, but this time I'm giving you 78 seconds. Just to give you a flavor, simulate the real life situation on boards, just to give you a flavor, how much is 78 seconds? You know, how much time do I have? Because you can't sit there. You can't sit there on the board exams and start dawdling and, you know, thinking, man, what's for dinner tonight? You know, man, you know, you have to think and focus. All right. All right. So here comes the first question. So here comes 2 questions, and hopefully get both of them correct. And the first one's not that difficult. The second one's a little bit harder. All right. So here is the first question. Ten seconds. Okay, and close the poll and broadcast the results. And see. Okay, thank you, Carrie. Okay, now, by the way, Lilia, please don't put the answer in the public chat pod because I'm trying to simulate the real life situation, the boards. You know, you know, when you take that board exam, you don't have help. You know, so if somebody happened to glance over to your 1 in 25, they well, what did she get to 125? You know, so please don't use the public chat pod to put an answer for a vignette that we're talking about. Okay? No, don't worry. That's okay. I'm excited that you're trying. Okay? I know I'm excited to try to help out other people. But for this case here, please don't put a response. But okay. Now use the selection pod for this. Okay? And so it looks like half the students who responded put option A, 1 in 25. And that is the answer, option A. Okay, let me move everything out of the way here and explain. Okay, now what happens here is they're saying the incidence of CF is 1 in 2,500. That's the prevalence of the disease in the population of Europeans. In fact, that's Northern Europeans. And that's like in Scandinavia and so on. And remember that cystic fibrosis is autosomal recessive. Again, they're not telling you in this question. You should know CF is autosomal recessive. So it's 1 in 2,500 in the population of Northern Europeans. And they're asking, what's the predicted incidence of carriers in that population? Not the worldwide population, in that population. So the answer is, so then it comes to Hardy-Weinberg. Again, they want to know about carriers. So again, here's the Hardy-Weinberg. P squared is homozygous normal. 2pq is the carrier status. Q squared is homozygous immune situation. And so again, forget about P squared. That's the normal situation. So we're either going to figure out carrier or we're going to figure out homozygous mutant. So what are they giving you and what are you trying to figure out? What they're giving you, they're saying the incidence of CF is 1 in 2,500. So they're giving you the homozygous mutant situation, it's 1 in 2,500. So then what we're trying to figure out is carrier status. So to figure out carrier status, we got to know what Q is, we got to know what P is. And so Q, we can figure out what Q is by taking the square root of 1 in 2,500. So again, as long as you can do square roots, then you'll get to the answer. And the square root of 1 in 2,500 is 1 in 50. By the way, since you don't have calculators on boards, to be safe, what I would recommend doing is if they give you a Hardy-Weinberg and you have to do a square root, take that number and multiply by itself again to go back to the original value to make sure you did the square root properly. Take the number you got, in this case here 1 in 50, Multiply it by itself again, and make sure you get 1 in 2,500. That's the square. So this is square root is 1 in 50. All right, so what are you trying to figure out? You're trying to figure out, that's the allele frequency, and try to figure out 2pq. And remember, p is that's the carrier frequency. That's what you're trying to figure out. The carrier frequency of CF in Northern Europeans. So the idea is, so we know that p is about equal to 1. For your purpose, it's 1. So all you got to do, there's the q. So it's 2 times 1 in 50. 2 times 1 in 50 is 1 in 25. That is the carrier frequency. Think of what that means. Think of the significance of this. Think of the significance of this. That means if you go to Northern Europe, you go to Denmark, you go to Sweden, Norway, Finland up there, you go up there to find a spouse or something like that. And suppose you are a female and you're going up there to find some guy named Born or Lars or something like that. Or if you're a female, you're looking for Haida or something like that. That the chances of you marrying somebody who's a carrier for CF is 1 in 25. If I was lecturing, if I went to Sweden right now and I was lecturing to 100 students, Swedish students, 100 Swedish students, 4 of the students, they may not even know it, but 4 of those students are going to be carriers for cystic fibrosis. It's a staggering number here. So a staggering number. As another example, I don't have, again, these carrier frequencies, and they usually do it on board exams for some regions of a country or a particular country. As another example, end of next month, end of June, I'm going to Saudi Arabia. I've been to Saudi Arabia the last 4 years. I taught at King Saud University in Riyadh and Al Faisal last year, and this year I'm going to Jeddah to teach over there, King Abdulaziz University, to the medical students there. But we go through this Hardy-Weinberg, and maybe if you know or don't know, but in Saudi Arabia, the incidence of sickle cell anemia is very high in a very localized part of Saudi Arabia, and that is on the eastern side of Saudi Arabia, that faces the Persian Gulf. And the incidence of marrying a carrier, if you go to eastern Saudi Arabia, the incidence, the probability, the carrier issue there for sickle cell anemia is amazing. 1 in 4. 1 in 4 Saudis that are in eastern Saudi Arabia are carriers for sickle cell anemia. The beauty of the Hardy-Weinberg, the beauty, and it woke a lot of students up when we went through that mathematics there. But I showed them the data, and we went through the mathematics, and they were, wow. We knew it was high, but we didn't realize it was that high. So again, these issues of the calculations. You know, I've been really blessed to be part of Kaplan Medical for 17 years, and I've been to a lot of places. I've been to a lot of places doing these live online, but I've also been domestically, a lot of cities in the United States here, overseas. Before I go, 2 weeks from now, actually, yeah, a week from now, I'm going to Grenada to go to SGU, come back for a week or two. Then I go to Poland, Poznan University. I've been there the last 2 years doing teaching to the medical students there. I love Poznan. The students are great there, as well as in Saudi Arabia. And then I don't know where else I'm going, but I think I'm going to Bahrain in August. So I go to a lot of places. So I'm and teaching students. We go through these Hardy-Weinberg. I usually try to get some disease that's in their country that might be high, and we go through these types of mathematics because it drives home the point. This is important to know. Okay, now we'll do one more question, and then we, yeah, it must be fun, but my wife. She says, I do too much traveling. But I said, but my students need me. I want to be with my students. My students, they need me. And she says, come on, get a life. You know, they'll be fine. I said, yeah, I know they'll be fine, but they need some guidance. You know, I know they need some guidance. And so anyway, but she's usually pretty good about all this kind of stuff. So, all right, here comes the last question. Then we'll take a 10-minute break. Then we're done with the chapter. Now, this next question is a little harder, but it takes thought. Ask, what are they asking me about here? Because it's going to take a little bit of thought. And you're going to ask, how can I get this done in 78 seconds? Well, as long as you don't dawdle, you should be able to get to the answer. Again, it's in your book. Don't read it. Don't look at the answer. Here comes the question. 78 seconds. Ten seconds. All right, then we'll close the poll and broadcast the results. Okay. All right, good. Yeah, Leo, well done. That was nice of you to say that. Leo, that was real nice. Natalie, you said if I go to Poland, what disease is prevalent there? We usually do Tay-Sachs because Poland is in Eastern Europe, and there's Tay-Sachs is in that lot of Tay-Sachs in that religious group of Ashkenazi Jews in Poland, as well as Eastern Europe. All right, now this question here, it looks about, so in terms of the results, 44 students put option C, and that is the answer, option C. All right, so, and a third of the students put option B. Okay, now let's see, now what are they talking about here? They're talking about a disease that nobody ever heard about. Most likely you, as well as most people, never heard about this disease called hyperprolinemia. But it is an autosomal recessive disease. It's an autosomal recessive disease. Again, I cannot emphasize enough. On board exams, you always underline, highlight, or make mental note, what is the mode of inheritance? That's the first thing you should always do if they give you that information. Make note of what the mode of inheritance is, because it's crucial. It's crucial to solving questions here. Now they're saying that the incidence or the prevalence of hyperprolinemia, a disease that most people never heard about, is 1 in 40,000. Now it says this man is a carrier for that trait, and if he mates with somebody from the general population, what's the probability that he and his mate will produce a child who's homozygous for the mutation involved? So the first thing we want to find out is, what's the probability that he marries a female who is a carrier for hyperprolinemia? What's the probability? Because he's a carrier. He is a carrier. And logically, he wants to know, what's the probability that I marry a person who's a carrier for the same disease? I certainly would want to know. All right, so here comes the Hardy-Weinberg. There it is. Hardy-Weinberg, p squared, 2pq is a carrier, and q squared, homozygous mutant. Okay, so what are they giving you, and what are you trying to figure out? How many times have I said that? So what they're giving you is q squared. Q squared is the homozygous mutant situation for this autosomal recessive disease, and we're trying to figure out the carrier status of that particular disease. So q squared is equal to 1. So to figure out the carrier status, we need to know what p is. We need to know what q is. Excuse me. And now to figure out what Q is, all we got to do is the square root of 1 in 40,000. So what's this? As long as you can do square root, the square root of 1 in 40,000 is 1 in 200. And just to be safe, just multiply it by itself one more time. You should get 40,000. All right, so now that we know what Q is, that's 1 over 200. We know, so that's the little frequency. The genotype frequency, 2pq, that's the carrier. That's what we're trying to figure out, is it's 2pq. And we know that p is about equal to 1. All right, so now that we know what Q is, all we got to do is substitute the Q for the value 1 over 200. So 2 divided by 100 is 1 in 100. That's the carrier frequency. That's the probability that he marries a carrier for hyperpolinemia. Again, what does that number mean? I mean, what does that number mean? That means somebody listening to me tonight in the audience right now. Somebody, one of you, or it might be me. Somebody listening to me right now is a carrier for a disease you probably never heard about. Never heard about. So it's amazing, huh? It's amazing that 1 in 100, somebody, if you have 100 people listening to me, somebody is a mutant, is a carrier for that particular disease. So why is the answer not A? And the reason is because they're not asking what's the carrier status. They're saying, okay, so if he marries somebody who's a carrier, and he's a carrier, what's the probability that they will produce a child who's homozygous for the mutation? All right, so here comes. Now, it's not hard math, but just a matter of thinking. The probability of marrying a carrier is 1 over 100. Now, let me ask you this. Of the female that he mates with, 1 in 100 probability of being a carrier. What's the probability? Let me see your answers here. What's the probability that her eggs will have the mutant gene? So if he marries a female who's a carrier, what's the probability that carrier's eggs will be mutated? Excuse me. That's right. Well done. That's right. So it's 1 and 2. So that would be the inherited mutated allele from the mother. 1 and 2 probability that the eggs, of all the eggs, 1 of every 2 will have the mutant gene. So why is the answer not 200 then? Option A. Why B rather? Why is the answer not option B? Let me see your answer. Before I give you the explanation. Why is the answer not option B? That's right, Gisela. Yeah, that's right. Because now there we go. You know why you got that? Because you're thinking. You're thinking. I could feel the vibes. The vibes are coming all the way over here to Kentucky. I could feel them. I could feel them. You're thinking. Oh yeah. You're thinking. I don't need to look at that first aid because I can think. That's right. Because it says he's a carrier, and because he's a carrier, what's the probability that his sperm will have a mutant gene? And it's 1 in 2. So therefore, then all you got to do is the mathematics, and it comes out to 1 in 400. There it is. There it is. Well done. Brilliant, everybody, because you can think. It's not hard. Not hard. It's not tricky. It's not hard. It's just a matter of figuring out what they're giving you and what are you trying to figure out. And just think your way through it. And mathematics is simple. So if you get a Hardy-Weinberg on board exam, something like that, and you're likely to just don't panic about the math. Just say the math is going to be simple. What are they giving me? What am I trying to figure out here? Just take it. Just use your mind. You got a beautiful mind. Just use it. Okay, I'm going to take a 10-minute break. Sorry, went a little overboard here, but I wanted to finish this chapter. So let's take a 10-minute break, and I will return in 10. Okay, now we're in hour 2 tonight, and we're done with chapter 2, and you know what's important there? Just one item, that's the Hardy-Weinberg and the use of it. And now we're getting to chapter 3, and chapter 3 is also just like chapter 1. Almost all, but there's some things that are minor, but almost all of it is a high yield chapter. You're going to see something that's coming out of chapter 3 with all these chromosome abnormality issues that Dr. Barone will also talk about from a pathology point of view, and what you're getting now is the biochemical point of view in these chromosome issues. So you're going to see my prediction. I cannot guarantee it, but I can almost guarantee you're going to see something about chromosomal abnormalities on the national boards. All right, now cytogenetics. Now, what does that mean? Cytogenetics means it's the study of microscopically observable alterations in the chromosomes. And there are two kinds of alterations of the chromosomes that can be noted. One alteration of the chromosomes are numerical alterations of the chromosomes. You and I have 46 chromosomes, but in the microscope, if I take some of your chromosomes of mine and lay them out, if there's not 46 chromosomes, if there's 45 chromosomes or 47 chromosomes, that's a problem, and that's a numerical alteration of the chromosomes. The other kind of alteration, and get ready for pathology, when you have an extra chromosome or missing a chromosome. Okay, and now the other one is a structural alteration of the chromosomes. And get ready for pathology there. There's always something going wrong with the biochemistry. So in terms of chromosomes, you're going to have numerical alterations. Nothing wrong with the structure. It's simply the number of chromosomes are abnormal. Or you can have 46 chromosomes, and what's the problem is there's a structural alteration of the chromosomes, and both of these situations can be observed under a microscope. Now, the idea of a microscope, if it's a structural alteration of the chromosome, a structure like a deletion or something like that, or a region of the chromosome is being deleted, how big would it have to be in order to see it in a microscope? And the answer is, it takes something like, it's not a small deletion. You're never going to see that in a microscope. In a chromosome, the structural alteration would have to be something like 2 million base pairs in length. So if a region of a chromosome that's missing or deleted or something like that, we will be talking about micro deletions coming up a little bit later. But the structural alteration would have to be something like 2 million nucleotides difference to be seen under a microscope. All right, so what's coming up here is we're going to be talking about the numerical alterations of chromosomes, then after that, the structural alteration of the chromosomes. But before we do that, we got to always talk about what's normal. I mean, you'll never understand what happens in pathology and what happens with these chromosome abnormalities unless you know what's normal first. So that's what we're going to do. One page of what's normal about chromosomes. All right, so the idea here is in these two panels, at the top panel, what the top panel is a metaphase drawing of the chromosomes. And the chromosomes are lined up 1 to 22, and then often on the lower right, doesn't have to be, but on the lower right would be the sex chromosomes, X and Y. Now, what you're looking at here, what these really are, these are one copy of each sister chromatid. Now, what the sister chromatid is, let me ask you, before I give you that, before I give you the answer to the sister chromatid, let me ask you this. When, in the cell cycle, see chromosome 1? In the cell cycle, when does chromosome 1 look like this? No, okay, so now be specific. I want you to be specific. When does the chromosome 1 look like this? Metaphase, that's part of it. G2 phase, that's part of it. Okay, here comes the answer. No, not G0. No. And here comes the answer. In the cell cycle, when your chromosomes look like this, at the end of S phase, all of G2 phase and the beginning of M phase, whether it's meiosis or mitosis. One more time. When do your chromosomes look like a sister chromatid? Remember, at the beginning of S phase, it's just one chromosome here. So it's not a sister chromatid. You have two chromosomes of a chromosome 1. And then they multiply. Let me do it one more time. So at the beginning of S phase, I have two chromosome 1s. So in S phase, the DNA is replicated in this chromosome. So now at the end of S phase, this chromosome is actually a sister chromatid where the green arrow is pointing to. The other chromosome 1 looks the same. It multiplied and it looks like this green arrow. So the idea is at the end of S phase, we have two sister chromatids. Two sister chromatids of each of the chromosomes. What you're looking at here is one of the two sister chromatids. These are a drawing here. Okay, one more time. So at the beginning of S phase, we have each of the chromosomes, we have two copies of them. One from the mother, one from the father. At the beginning of S phase. Not G0 phase. There's no replication going on in G0 phase. But the idea is the beginning of S phase. Then what happens is the DNA multiplies. At the end of S phase, that particular chromosome over here, that particular chromosome over here now looks like this. The other chromosome 1 also multiplied, and it looks like this as well. Here. Okay, and that's at the end of S phase. It looks like that in G2 phase, and it looks like that at the beginning of M phase. And then in M phase, eventually, the sister chromatids will then separate into the single chromosomes. But in the sister chromatids, there are actually two chromosomes. Okay, now the idea here is in these sister chromatids, where they're joined together at the end of S phase, that's called, what's it called, by the way? Where they're joined together. See over here in this X chromosome, for example, here, where they're joined together. Again, this is a chromosome here, and here's a chromosome there at the end of S phase. So this, like, for example, this chromosome may have multiplied by another one to form this one. So now, but they're still held together. What's that called? That's right, it's called the centromere. So the centromere is where they're being held together at the end of S phase. And then relative to the centromere, then the shorter arm is called the P arm, and the longer arm is called the Q arm. So the shorter arm is P arm, and the longer one is the Q arm. By the way, I know I don't want to. This might be obvious to you, but the P and the Q here. We're going to hear a lot about P's and Q's in this chapter. This has nothing to do with the Hardy-Weinberg. Remember the Hardy-Weinberg, the P squared. Remember it's Q. That's something different. Don't get that confused. That P and the Q there with this P and Q arm business here. So they're not the same. P stands for petite and all that kind of stuff, so don't get them confused. It's nothing to do with the Hardy-Weinberg. Okay, now, so there's the centromere here. By the way, what's the end of the chromosome called again? What's the end of that chromosome called? The end of the chromosome, what's it called? That's right, it's called the telomere. That's right, well done, well done. It's called the telomere in the middle part or somewhere in the chromosomes where they're held together. It's called the centromere. Now, another item that you must know, and that is there's a class of these chromosomes that are called the acrocentric chromosomes that you must, and there are 5 of them, and you must know them by heart. And they are chromosome 13, 14, and 15, and chromosomes 21 and 22. And what the acrocentric chromosomes are, these are the chromosomes that have those very short P arms. Look at chromosome 13, 14, and 15. Chromosome 21 and 22, they have those very tiny P arms. Chromosome 13, 14, 15, and chromosome 21 and 22. Later on in the chapter, I'll explain to you why it's important for you to know the acrocentric chromosomes. Major pathology coming up. Okay, so these are the acrocentric chromosomes. Now at the bottom panel, that's the real life situation. Now, where the chromosomes are laid out in numerical order. By the way, they're laid out in numerical order. This is chromosome 1, numerical order in terms of the size.
Chromosome 1 is larger than chromosome 2, is larger than chromosome 3, larger than chromosome 4, all the way to the smallest chromosomes, and that's chromosome 22. So in the karyotyping that goes on, the chromosomes are laid out based on size, starting with chromosome 1 to chromosome 22. Now, another item here. A lot of students never thought of this. So it may be you. You probably never dawned on you. This is really what's happening. So let me be careful here to explain, straighten it out if it never occurred to you. If I were to ask you, in reality, what is this thing over here? See where the green arrow is pointing to? What is this? What this is? What is this? I mean, is it a chromosome? Is this a chromosome? Is this a chromosome? That's right. That's right. It's not a chromosome. It's two chromosomes. What it is, it's a sister chromatid. There are actually two chromosomes here, chromosome 1s, that are held together at the centromere. And in other words, suppose this is chromosome 1, sister chromatid. And at the end of S phase, it looks like this, G2 phase and MM phase. So in metaphase, that's where the chromosomes are actually being stained. But when they get stained, what happens is, notice here the P arms, there's daylight between them. The Q arms, there's daylight between them normally. But what happens is when they're being stained, the daylight, the P arms collapse with each other, and the P arms collapse with each other. So a lot of students think this is one chromosome, and it isn't. It's two chromosomes. So what in reality, when you do karyotyping, it's actually two copies of each sister chromatids. So in other words, there are actually four chromosomes 1s here. Four chromosome 1, 4 chromosome 2s, 4 chromosome 3s, and so on. Because these are sister chromatids, and each sister chromatid has 2 chromosomes there. So again, sometimes people never thought of that, but just to make sure we understand in reality here. So, okay. Now another item here is that at the bottom right of the figure are the sex chromosomes. So, so all the other chromosomes are the autosomes, and the sex chromosomes are the X and the Y. Look how big the X chromosome is compared to the Y chromosome, and how big it is compared to most of the other chromosomes. So there's a lot of genes on the X chromosome here. Okay, and then the other ones are called the autosomes, chromosome 1 to 22. Again, that relates to chapter 1. When we talk about autosomal dominant diseases, autosomal recessive diseases, what does that mean? That means the gene of interest, like in PKU or sickle cell anemia or something like that. That means the gene of interest is located on one of the autosomes. And whereas they say X-linked, that means the gene of interest is located on the X chromosome here. Okay, now another item here is there's another term we need to attend to, and that's the term called euploid. Euploid is the exact multiple of the haploid situation. So in other words, in your gametes, whether you're female or male, in your sperm or egg, you have 23 chromosomes here, and that's the haploid situation. But in all the other cells in the body, you are diploid because you have 2 copies of each of these chromosomes here, well, except for the sex chromosomes if you're male. Okay, so euploid is an exact multiple of the haploid situation here. Okay, so now so in this chapter, when I show you, if you were to visualize a karyotype or something like that, and they say 46 chromosomes, in reality, you have to keep saying to yourself, really it's not 46 chromosomes, it's actually 92 chromosomes. So if I say the normal karyotype is 46XX, in reality it's they're referencing the sister chromatids, not the chromosomes. All right, now what's coming up, so this is all the normal situation, Okay, so now what's coming up next is the numerical chromosome abnormality situations. Numerical chromosome abnormalities. Now the normal, so me and you, if we're normal, we are diploid. And if I'm a male and you're a female, the karyotype might be shown or listed as 46, XY. That's the normal male karyotype, whereas the normal female karyotype is 46, XX, and that is the normal situation. Now, but now in the numerical chromosome abnormalities, then there's something wrong with the chromosome number. The structures of the chromosomes are fine. There's something wrong with the number. And if and there's two subgroups of the abnormalities, and these are called aneuploidies. So aneuploidy means not 46, not 46. So the idea is, could be 47 or something like that, but the idea is not 46. And of the autosomes that can be, you can have an extra chromosome of one of the autosomes. There's a major one. Everybody, if I were to ask what's the best example of an autosomal aneuploidy, everybody should be writing, what's the best example of an let me see, best example of an autosomal aneuploidy. Again, aneuploidy means not 46. That's right. Everybody should say Downs, trisomy 21, Down syndrome. And what happens there is that they have 47 chromosomes, because now it's an aneuploidy because it's not 46. And this could be a male, and on board exams, if you see a plus 21, that's telling you that there's an extra chromosome 21 in this male for a total number of chromosomes of 47. And this would be a female under the same circumstance. So they might actually list the karyotype this way, or actually show you a karyotype like you saw on the previous page, but then you look at chromosome 21 and you see there's an extra chromosome 21 there. If they ask you what disease is it, you say that's Down syndrome. Now, as everybody knows, well, first of all, Dr. Barone, all these particular aneuploidies coming up here, all the pathology, clinical manifestations, save it for Dr. Barone, because he's the expert on the clinical issues there. So, so all that, the issues of what the people look like and all this kind of stuff, I'll leave it up to him. He has great mnemonics for those type of things. But here, the issue is in terms of Down syndrome, as we talked about the other day, it is the number one most common cause of mental retardation. And the incidence of Down syndrome is very high, very high. And it's one, and generally it's about, if a female on the x-axis here, the maternal age, if a female is 30 years of age or less, the incidence of Downs is still pretty high. It's 1 in 1,000. The incidence is very high of Down syndrome. But notice as a female gets older, as she reaches 35 and so on, the incidence of Downs child that she will have will start to increase, and then when she then becomes exponential, and then when she reaches age, let's say 42 or 43 on this graph, then the incidence of Down syndrome is 1 in 50. So as a female gets older, then the chances of having a Down child is greatly increased here. And it has a lot to do with the fact that the eggs of a female are made at birth, and they can be damaged and things along that line. But I don't worry about the details here, what's causing this to happen. But just know that the female, and everybody knows this, that the female, as she gets older, will have a greater chance of having a Down syndrome child. Okay, now again, Dr. Barone will talk about all of the pathologies. Now, the next one to talk about is another autosomal aneuploidy, is trisomy 18. That's Edwards syndrome, and so on board exams, you might see the karyotype 47, XY, plus 18, and what that's referring to is that is a male who has an extra chromosome 18 for a total number of chromosomes of 47. So this would be a male, and that would be a female under that circumstance. Now again, Dr. Barone will talk about the pathologies associated with Edwards syndrome. But remember, they have the fingers fold and so on. The tragic disorder is Edwards syndrome. Not Cri-du-chat. Cri-du-chat is not this. No, don't. Cri-du-chat has nothing to do with this. All right, now coming up, now the third one to talk about here is trisomy 13, which is called Patel syndrome. So you might see it by either name, Patel syndrome or trisomy 13. And the way they might show you the karyotype, or they might just say a patient has this karyotype, 47, XY, plus 13, and you say, well, that's a male who has an extra chromosome 13 for a total number of chromosomes of 47. And so, this would be a female under the same circumstance. So these are the autosomal aneuploidies here. Now, now let me ask you this. What can you tell me about the other autosomal aneuploidies? What can you tell me about trisomy 16, for example? What about the other autosomal aneuploidies? That's right. That's right. That's right. Well done, everybody. That's right. That they are lethal. They are lethal. They're incompatible with life. Now, if I were to ask you why, I mean, so you have an extra chromosome around. I mean, you can memorize all you want, but I'm going to try to get you to think. Okay, you got a beautiful mind. Just use it here. Just use it. Okay. Why is it that if you have an extra chromosome around in a cell, that it's going to be lethal? It'll never survive in utero. I mean, well, why is it lethal? So there's an extra chromosome in the nucleus. Okay, so what's the problem? Right? What do you think? What do you think? Yeah. By the way, from the first trimester abortion, trisomy 16 actually is the most common first semester trisomy. By the way, I doubt the National Board would ask that, but it might. The most common cause of first semester trisomies is of first semester abortions is trisomy 16. But why is it? Gisela, we're talking about autosomes, not sex chromosomes. Hey, sex chromosomes are coming up shortly, Gisela. Okay, so not XXY. No, okay. So those are be specific. Autosomes. We're talking about chromosome 1 to 22. Okay. Okay. All right. So now the idea is, no, it's okay, Gisela. At least you're trying. You're thinking. You're thinking. You're my type of person. If you think, if you memorize it, save it for other courses or other disciplines or other companies. But if you're a thinker, you're my type of person. But now to try to think your way through this. So why is it if you have an extra chromosome, another autosome around, like trisomy 16, why is that lethal? Organs don't develop properly. It all gets messed up. Yes, Siad. No lionization. Lionization, that's sex chromosomes. No, that's sex chromosome. We're talking about autosomes. And okay, there we go. And the reason is, remember, when you have two chromosomes, when you have a particular chromosome, let's say chromosome 1, chromosome 16 or whatever, and you got two copies of the chromosome. And in each chromosome, you have genes that need to be turned on. And as we talked about yesterday, when you turn on one gene in one chromosome, you turn on the same gene on the other chromosome that you inherited from your mother or your father. Okay. And so then those genes are turned on, they make protein, and then the cell works and all this kind of stuff. But now if you have another chromosome around, then instead of having two copies of that particular enzyme being made, two copies of the gene, making a certain number of enzymes. Now you have an extra gene around, and it's making even more enzymes. And more enzymes in the cell, cell doesn't like it. And so the number of enzymes in a cell are tightly regulated. You just can't keep having extra enzymes around without thinking it's not going to cause havoc to the cell, to the organ, to the body. That's why it's lethal, because having an extra chromosome around, there's too many enzymes being made from those extra genes on that particular chromosome. And the cell and the organ and the body can't survive. All right, now, so that's why the other trisomies. Now look at these other ones, these three here. They can survive. But notice these are very small chromosomes, chromosome 21 and 18. They're pretty small chromosomes. So the number of extra genes are not as many compared if you had chromosome 1, which is a lot of extra genes that are going to be turned on to make extra proteins. But even here, Downs, Edwards, and Patel, they got serious pathology because there's extra genes in they have one extra chromosome that's making more enzymes. And more enzymes in the cell, that's a problem. All right, now one more question here. What can you tell me about the autosomal aneuploidies that are monosomies, where you're missing a particular autosome. We can tell me about that. Let me see your answers here. The autosomal aneuploidies in which they're missing one of the autosomes. Missing material. Tariq. No. Tariq, that's Klinefelter's is a sex chromosome aneuploid. Everybody wants to talk about sex chromosomes. No, get that out of your mind. Get that sex stuff out of your mind right now. We're talking autosome. Listen to the words. Autosomal monosomies. Which chromosomes 1 to 22. Not the sex chromosomes. But the idea is that if you're missing one of those autosomes, what can you tell me about them? And I thought I saw it earlier, and the answer, that's right. And the answer is that it's lethal. All autosomal monosomies are lethal, incompatible with life. Now, why is that? Again, the biochemistry is easy to explain. If remember, normally we have two sets of chromosomes and the genes in those chromosomes are making RNA, making enzymes. And the amount of enzymes that are going to be made got to be tightly regulated. But now if one of the chromosomes is missing, then you're getting half the number of enzymes that's supposed to be made from that particular chromosome. And that's not enough enzymes. And therefore the cell doesn't like it, and the cell dies, the organ dies, the body dies, incompatible with life. It makes sense. That you must have, that's why they're all lethal, because you're not having enough enzymes in the cell, in the monosomy situation. All right, now everybody wanted to start talking about sex chromosomes. All right, let's go to sex chromosome, aneuploidies. Now the sex chromosome aneuploidies, there are two major ones you need to know, and the first of these is, I forgot who mentioned that earlier, That was the Klinefelter. Klinefelter. Now Klinefelter's, that's right, Muskin. I should trademark it. You're going to hear it a lot, by the way. And Klinefelter. Now, Klinefelter's is it's where they have an extra sex chromosome around, and it's 40. So therefore, its total number is 47, XXY. And the important feature there is that the Y make that person always a male, and it's expressed after puberty in Klinefelter syndrome. Now, again, the pathology will be discussed by Dr. Barone, but it is a sex chromosome aneuploid, an extra sex chromosome. But in my opinion, even more important disease for boards is sex chromosome aneuploidy. Everybody likes to say that, and that's Turner syndrome. Now, Turner syndrome is, you need to know Turner syndrome for multiple reasons. That's why it's so high yield of a disease for board exams. Just like we talked about earlier, and we will talk about Tay-Sachs disease. It's used to illustrate many things. But here's another example. You need to know Turner syndrome, not just for the pathology. You need to know it for other reasons, because it illustrates several different principles in genetics. And one principle that it illustrates is it is the only monosomy in which a person can survive, either autosomal or sex chromosome monosomy. And it is a sex chromosome monosomy. Where they're missing one of the sex chromosomes, all the other autosomes are normal. So the total number of chromosomes is 45, and because it's a monosomy, it is it's the X chromosome here. So the Turner syndrome is used to illustrate the principle of the only way a person can survive a monosomy is Turner's. Now, what's another reason why you might see Turner syndrome on a national board? What's another reason why you might see Turner syndrome on the board exam? Why? Anybody know? No bar body? No common. I'm not talking about the pathology, not the pathology. And the reason, and the other thing you need to know about it is, it's used to illustrate. So some of the cells in the Turner's patients has the karyotype 45X, and other cells, which is a monosomy, and other cells in the body can have the normal karyotype 46XX, for example. And what is the name that this is called. What's this called when some of the cells in the body have it? Sorry about that. What's the name of that? What's the name of the what's this called in genetics? Where there's a some cells have 45 chromosomes, other cells have 46. That's right. It's called mosaicism, and this is tissues with genetically different cells. So mosaicism are tissues with genetically different cells. Some cells in the body have the normal karyotype, 46 chromosomes. Other cells in the body have the 45X, and that illustrates the principle of mosaicism. Yeah. All right, so that's another reason. A second reason you need to know Turner's from a genetic point of view is the monosomy issue as well as mosaicism. Now, if I were to ask you, you might see Turner syndrome on boards for yet another reason in genetics. Does anybody know? Usually when I ask, most people don't get it right. What other reason in genetics that you might see Turner syndrome used to illustrate what principle? Anybody know? Not the ring. Not the ring. Good try, Omar. Good try. At least you're trying. At least you're participating. Pleiotropy, no. And the answer is that Turner syndrome is used to illustrate the principle of the isochromosome. An isochromosome. That's a structural alteration of the chromosome. Turner's patients usually have a numerical alteration of the chromosomes. But you can have Turner's patients which the total number of chromosomes is 46. But the problem is one of the X chromosomes is structurally altered, causing Turner's syndrome. And that structurally altered X chromosome is called the isochromosome. That's coming up later on in the chapter. Don't worry, I got your back. I'm covering it later on. But right now we're talking about numerical alterations of chromosomes, and later and then later on we'll talk about the structural ones. And you'll see Turner's again. I'm telling you, they love Turner's syndrome for the boards because it illustrates many principles. Here, now in embryology and so on, what's causing? If they were to ask you, what is the basis, the molecular basis for having an extra chromosome or missing a chromosome, and all you need to know is this: numerical chromosome aneuploidies are generally caused by something called disjunction. Normally, when sister chromatids get into M phase and in meiosis, they go through anaphase 1 and anaphase 2, and they disjoin either in anaphase 1, anaphase 2. That's all the stuff talked about in embryology. They disjoin. But the problem is, what could happen is that there might be a failure to disjoin of one of the sister chromatids. And when it should. If it doesn't disjoin when it should, it can cause the extra chromosome to go into a gamete, which can then be fertilized. So, but in your book, there's a couple pages that shows how it happens, how non-disjunction happens, and all. Forget about it. I think that's low yield, so we're going to skip over that. So everything you need to know about numerical chromosome aneuploidies is right here. It's as simple as I can get. I don't want to make it complicated, but here it is right here. Don't worry about how you generate non-disjunction. That's going to be much, much lower yield. So now we're going to get into the structural alteration of the chromosomes. And it's going to take a lot longer time to go through that. Now, the structural alterations of the chromosomes is, there's going to be four sentences coming up here that set the stage for the rest of the chapter. 4 sentences. So the idea is, so your chromosomes are sitting in the nucleus. And what can happen is, as they sit there, there can be some structural problem that can go on. Some chromosome might lose a piece or something like that. And there's a term called balanced alterations of the chromosomes. 90% of chromosomal abnormalities they're called balanced alterations. Balanced means if I have one chromosome here and another chromosome over here, if a portion of this chromosome goes onto this other one, and a portion of this other chromosome goes over here, that's a structural alteration. In other words, if the P arm of one chromosome goes on to another chromosome, and then the P arm of the other chromosome goes back in the other one, something like that, rearrangement, that's called balanced alterations of the chromosomes. There's no loss or gain of genetic information. Excuse me. The location of those genes will be different. I mean, it may not be in the particular chromosome they're originally on, but the genes are still there. So these are called balanced alterations of the chromosomes, where a piece of a chromosome goes onto another one. So there's no loss, there's no gain of genetic information. Now, unbalanced alterations, which are much less, 10% of the structural alterations, result in loss or gain of genetic information. Here. And so unbalanced alterations result in, so if somebody inherits a piece of DNA, a chromosome that's missing a region, that's going to be unbalanced alterations because it's going to result in the loss of genetic information. And now, another item here. Those structural alterations that occur in the germline can be transmitted to the offspring. Either balanced or unbalanced alterations. If they occur in the germline, there'll be nothing wrong in that individual. There'll be nothing wrong in the father or the mother if it's happening in the germline of the father or the mother here. But if it happens in, so if it happens in the germline, no problem in that person. But the problem can, as you'll see coming up shortly, but if the problem happens in germline, that can be transmitted to the offspring. The offspring will have the problem. Okay, now those structural alterations that occur in a somatic cell can cause cancer. Matter of fact, well over 100 different cancers are known to be due to structural alterations of chromosomes that occur in somatic cells. And the pathology will occur in that individual. Okay, one more time. So if you have a structural alterations, if a structural alteration occurs in my germline, nothing's going to be wrong with me. I'm fine. But the problem is it's going to be transmitted to my offspring, and my offspring will have the problem. But if they have a structural alteration of the chromosome in a somatic cell, like a skin cell, something like that, that's where the problem can arise, because it can give rise to cancer here. Okay, now let's look at the time, and it's 8 minutes to the hour. So what we're going to do is we're going to take a 10-minute break, and then I will be back and then get into this all-important topic called reciprocal translocation. You must understand reciprocal translocation. So let's take a 10-minute break, and I will be back.
can't stop it on this.
I know.
That's why I called
Okay.
my dad on my way over. And said he was
All right. There we go. There we go. All right. There we go. Back. Too many buttons to press over here. Okay, so we're back here, and so we're now talking about the structural chromosome abnormalities. And now what's coming up next is this issue of reciprocal translocation. It is extremely high yield because it's the cause of many types of diseases coming up, cancers and so on, reciprocal translocation. Now, first of all, we're going to talk about what's normal. Now, what's normal is, suppose, to color code things, suppose we have two chromosome 2s, and we have in red, and we have two chromosome 8 in blue. Now, what normally happens is that we can have crossing over between two homologous chromosomes. So, the tip of this chromosome 2 can come over here. The tip of this chromosome 2 can go over there, back and forth. And same thing with the Q arms. That's normal situation goes on. That's normal crossover. We'll talk more. We'll talk more about that later on in chapter 5. But that's crossing over between. I want to emphasize between homologous chromosomes, the same chromosome number. Same chromosome number. It's homologous chromosomes. So the same thing with chromosome 8. The tips of the chromosomes can cross over. That's the normal situation. All right. Now the idea here is in pathology. So by the way, you might ask. Well, when the chromosomes cross over, won't that disrupt genes? You know, when you take one portion off and put it onto another portion of a chromosome, won't that mess up a gene or whatever? And the answer is generally no. And the reason is because that's why it's another reason why you have so much junk DNA. You have so much junk DNA because you have crossing over between chromosomes, and it won't cause any problem. The only pathology that I'm aware of that's high yield for boards that's due to unequal crossover is what? And from pathology, what's the best example of a disease that's due to unequal crossover between two homologous chromosomes? Two homologous chromosomes, you get unequal crossover. They don't cross over like they should. It's unequal. Which is the best example? Not 922. No, that's coming up next. No, not Cri-du-chat. People want to keep saying Cri-du-chat. That's coming up later in the chapter, but it's not Cri-du-chat. And the answer is, people not Downs. Downs is a numerical. Hey, Natalie, Downs is numerical, not a structural alteration here. I think you're thinking Robertsonian, but that's not. No, that's coming up later. That's also coming up later. And that's between non-homologous chromosomes. And the answer is, again, it comes to the question one more time. What's the best example of a disease in pathology that's due to unequal crossover between homologous chromosomes? And the answer is alpha thalassemia. Alpha thalassemia is the most common cause of alpha thalassemia is the unequal crossover between whatever chromosome number the alpha globin gene is located. I can't remember. That's not a big deal to know the number of the chromosome. But two homologous chromosomes, you get the unequal crossover where the alpha globin gene happens to be located, and that's causing the pathology. The best example of unequal crossover between homologous. The key word there was homologous chromosomes. But now that same word, the key word homologous, now put the word non-homologous. And that's what reciprocal translocation is. Reciprocal translocation, by definition, is chromosomal rearrangement between non-homologous chromosomes. So here are two non-homologous chromosomes. Here's chromosome 2 here, and here's chromosome 8 there. And what can happen is the P arms or the Q arms between these two non-homologous chromosomes can cross over. They can just cross over with each other. And by color coding, notice how the P arm, portion of the P arm of chromosome 8 is shown in the blue, is now located on chromosome 2. And the P arm of chromosome 2, a portion of it, is now found on chromosome 8, shown here in the red on the blue chromosome 8. And now the Now the idea here, the concept here is the karyotype you might see on board exams is you might see it where the total number of chromosomes are still 46. There's no loss, there's no gain of chromosomes. So this could be in a male or in a female, and what the T and so on stands for, the T stands for translocation of the P arm of chromosome 2 with the P arm of chromosome 8. That's exactly what you see here. So this would be an individual. The rest of the chromosomes are not shown there. Chromosome 1, chromosome 5, they're not shown here. So the total number of chromosomes is 46. XY male and so on, and female, and the T stands for translocation of the P arm of chromosome 2 with the P arm of chromosome 8. Now let me ask you this. If this occurred in me, would there be any problem in me in terms of pathology? So in one of my cells, if this happened to me, would there be, let me back off a minute. If this happened in my germline, let's look at the germline first. If this happened in my germline or your germline, reciprocal translocation, would there be any problem? That's right, and there would be no problem. And why is that? Because you still have 2 copies of the genes. No problem in an individual if it occurs in the germline. You still have 2 copies. Whatever genes were located over here now are located over here, as you see right here. And whatever genes were located over here are now found on chromosome 8. So the locations of the genes are different, but it still are diploid. We're still diploid for the number of genes. There's no loss or gain of genetic information. So in my germline, there's no problem in me if it happened in me, because I still have 2 copies of the genes. And if it happened to me, I'm called a reciprocal translocation carrier. Or anybody, if it occurs in the germline, that person's called a reciprocal translocation. Look at the words. Reciprocal translocation, we just talked about, and he's a carrier. Okay. But the problem can present in the offspring, because now if this is going on in a germline, then what's going to happen? Now here is the problem. So the problem is in the germline when you're forming sperm or eggs. What can happen is this chromosome 2 can go into one sperm and everything is fine. But this chromosome 2 can go into another sperm. Remember, in the haploid situation, remember all the meiosis? You have to know embryology to understand what I'm getting at here. But in gametogenesis, when you form the haploid sperm or eggs, this chromosome 2 will go in half of the sperm and the eggs, and everything will be fine. And this abnormal chromosome 2 will go in the other half of the sperm or eggs here. And that's a problem, because now that person's missing these genes. Whatever were in the P arm of chromosome 2 are not there if it happened to go in a particular sperm here. And the same thing with the same situation goes on with chromosome 8. And so that's in the offspring. So again, if it happens in my sperm or eggs, and that happens to fertilize a normal normal egg, then in the zygote, likely going to be incompatible with life. Because it's unbalanced translocation, because either not enough genes or too many genes are being generated here, and that's the problem. Too many loss or gain of genetic information. But there's no problem in me, it's in the offspring. Okay, so now this is if it happens in the germline. Now what about if this reciprocal translocation occurs in a somatic cell? If it happens in a somatic cell, in me, for example, if it happens in my skin cells or something like that, then that can give rise to cancer. And there are about 100 different kinds of cancers, about 120 or 30 last I heard, that are due to reciprocal translocations. So again, this is pretty frequent that it happens. And of those cancers that are known, 2 of them you must know, that we're going to emphasize here. 2 of the most important of the cancers caused by reciprocal translocation. Remember, some cancers are caused by tumor suppressor gene loss, and later on we'll talk about oncogenes in biochemistry. But here is reciprocal translocation causing cancer. Okay, now the idea here is, and then the first of these two, everybody's going to have to say, is CML, chronic myelogenous leukemia. Now there's a diagram coming up that's not in your book, and just took this off the internet. But the idea is and these chromosome numbers are absolutely important to remember. This is chromosome 9 on the left side, fairly large chromosome. This is chromosome 22, very small chromosome. And on the Q arm of chromosome 9, there's a gene called the ABL gene. On chromosome 22, there is a BCR gene. And what happens is in reciprocal translocation, remember, normally these chromosomes will not cross over because they're not homologous. But it can happen, and so the Q arms can cross over, and when it crosses over, what generally happens is then just so happens there's abnormal translocation where the ABL gene on chromosome 9 now fuses with a BCR gene on chromosome 22, and when we get that fusion over here, that's, everybody knows, that's called the Philadelphia chromosome as the product. When if it happens in a, for example, in a rapidly growing cell like a white cell, here, causing leukemia. All right, now let me ask you this. Okay. So you get ABL gene now fused with the BCR gene. Well, so what? So what? What's this got to do with cancer? So you got two genes fusing with each other. So why does a person come down with chronic myelogenous leukemia? How does the biochemistry explain the pathology, which will then explain the pharmacology that I'm going to talk about here? And that's right. And the reason what happens is the fusion of these two genes results in a continuously active tyrosine kinase activity. Okay, now what does that mean? Okay, well, so it results in a continuously active tyrosine kinase. Well, so what? What's that got to do with cancer? Okay, here comes the biochemistry integration with the pathology. And be ready for the farm. The idea is, we'll talk about this later on in metabolism. Once we get to insulin and the growth factors, how do they work? What happens is on cells in G1 phase, in cells in G1 phase, there is a class of receptor called the tyrosine kinase receptors. The class of receptor, tyrosine kinase class of receptors. And the tyrosine kinase class are never active. They're sitting there, the receptors sitting there doing nothing. But then when you secrete the hormone, like epidermal growth factor or whatever it is, and then from Dr. Wilson's review, then the hormone will bind to the tyrosine kinase receptor. The receptor now becomes activated, and then it signals the cell, signals the cell to go into S phase, multiply the chromosomes, and then grow and divide. Why? Because the hormone activated the receptor. But the problem here is, by fusing of these two genes, the receptor on the surfaces of these white cells, the receptor is now active in the absence of the hormone. There's no hormone being secreted, yet now the receptor is activated, and when it's activated, it's going to cause these cells to grow and grow and grow, and there it is. That is the chronic myelogenous leukemia, as simple as I can explain. You must know the biochemistry first. The biochemistry says the tyrosine kinase is never activated unless you secrete the hormone in the cells in G1 phase. But now it's active in the absence of the hormone because of the fusion of the genes, and that's the problem. All right, now let me ask you this: in pharmacology, what's the name of the drug commonly used to treat chronic myelogenous leukemia, and why do you give the drug? That's right, it's called imatinib, also called Gleevec, commercially known as Gleevec. So why do you give imatinib? Why? There you go, that's right, just makes sense, because by giving imatinib, it's an inhibitor of tyrosine kinase activity. Excuse me. So by giving imatinib, you inhibit the enzyme, the receptor from working in the absence of the hormone. and therefore the leukemic cells, the myelogenic leukemic cells will not grow anymore because the receptor is no longer active in the presence of the drug. It can't signal the cells to begin multiplying. There it is. That is as simple as I can explain why you give that drug in pharmacology to treat that disease in pathology, the biochemical basis of it. Now, the other disease, major disease, again, Dr. Barone's certainly going to be talking about CML. Be ready for it. And you know the biochemistry right here. Now, the idea next is, another example is Burkitt's lymphoma, the other major one you got to know for board exams. There are other ones as well, like acute promyelogenic leukemia, AML, subtype M3, those type of things. But for the highest yield cancers that illustrates the principle of reciprocal translocation is CML and Burkitt's. Now let me ask you this. Now before we get to, before I explain the pathology, let me tell you the normal situation. You always got to know the normal biochemistry first to understand the pathology. You can memorize the pathology, sure, but you'll never understand it unless you know what happens normally first. Try it out sometime. Try talk about some disease or whatever, even diabetes. You'll never understand it unless you know what's supposed to happen normally first. All right, let me tell you the normal situation of a particular protein. There is a protein called c-Myc. And it is a proto-oncogene, and we'll talk more about proto-oncogenes later. But c-Myc is a normal protein, and it just so happens it's a specific transcription factor. How often are we talking about those in Chapter 5? But here's another one you need to know. This is c-Myc. It is a specific transcription factor. It's located in specific tissues. And what it does, amazingly, when it's turned on, when c-Myc is turned on, it then binds to enhancers and regulates the expression of 15% of all genes in that particular cell. Amazing. I mean, that's, the numbers are incredible here. And to prevent this this c-Myc from always turning on these genes, it's going to be highly regulated. So you only turn it on when you need it. And so therefore it turns out it has a very short half-life of only 30 minutes. It's one of the shortest half-lives of a protein that's known. 30 minutes. It's made, does its thing, and destroyed by the ubiquitin proteasome complex that we talked about the other day. So it's made, works, does its thing, turns on certain genes, and then it says, man, I'm tired. Enough is enough. I'm tired. I'm gonna misfold, and it misfolds, and quickly the proteasome gulps it up and chews it away. There it is. That's the normal situation. Now, what happens in Burkitt's lymphoma? There's reciprocal translocation of 2 chromosomes. Let me see your answers here. Which chromosome? What's the chromosome numbers here? You need to know for Burkitt's lymphoma. These ones are important. That's right, and these ones are chromosome 8 and 14. Now what happens is when you get reciprocal translocation there, then just so happens that 2 genes fuse. One gene is the immunoglobulin heavy chain. That's really not the problem. The problem is the other one. It's right near the c-Myc proto-oncogene. And so now they fuse, the genes fuse, and they make a fusion protein, and portion of that fusion protein is c-Myc. And so now it turns out it's not turned over in 30 minutes. c-Myc is sitting around for hours and hours and hours. As it sits around, it turns on more and more genes. And then things start to grow and grow and grow. That is Burkitt's lymphoma. There it is right there. It's just simply that the c-Myc proto-oncogene, heavy chain, it's not turned over like it should be in 30 minutes. And therefore it sticks around longer. It's stabilized. And causes the cells to continue to grow because the genes that are being turned on are involved in cell replication. There it is. Okay, now again, when Dr. Barone is here, I'm certainly going to talk about Burkitt's lymphoma. So these are the 2 most important ones. Again, there are other ones as well, but certainly know these 2 as illustrators of the principle of reciprocal translocation. Okay, now what's coming up next is another kind of translocation. And also, very high yield. I'll explain why. And this is called, as Natalie mentioned earlier, that's called Robertsonian translocation. Now, what Robertsonian translocation is, and you must know it, and this is it involves the loss, and that's of the short arms. Remember the P arms of the acrocentric chromosomes. Remember in the first figure at the beginning of the chapter, I said to you, you need to know the 5 acrocentric chromosomes. Again, there's no memorizing. The chromosome 13, 14, 15, and the 2 smallest ones, chromosome 21, 22. So again, the 5 of the acrocentric chromosomes. And here are 2 of the 5, chromosome 14, chromosome 21. Now it says what's going to happen is the P arms can come off. And then what can happen is, in Robertsonian translocation, the Q arm of chromosome 21 might now fuse with the Q arm of chromosome 14. And there it is. By color coding, you see here's chromosome 14 and here's chromosome 21. So the karyotype, by the way, and so you're losing the P arms here and the Q arms just fuse. But the important feature is no essential genes are lost. And the reason is because the important genes on the acrocentric chromosomes are on the Q arms, not the P arms. So you can lose the P arms, no big deal, no pathology, and the Q arms will fuse. So no essential genes are lost. Now the karyotype would be something, but and the reason is, notice that they were still diploid. We are still diploid on the number of genes on chromosome 21 and 14. So whatever genes are located here, you got 2 copies of them, and whatever genes are on chromosome 14, they're right there. So we're still diploid for the number of genes. No problem here. That's why it's really not a problem. Okay. So in this situation, the karyotype would be 45. This would be a male, and this would be a female under the same circumstance. So 45 chromosomes because there's a loss of one of the chromosomes total. Let me put it another way. There's a loss of one total chromosome. Okay, but even though but it's a structural alteration, but there's a loss of total number of chromosomes are 45. Okay, now and then it says here. So if you see 45XY, that would be a male who has a total number of chromosomes of 45. And the reason is because he has lost chromosome 14, lost chromosome 21, but gained the plus, but gained the translocation of the Q arm of chromosome 14 with the Q arm of chromosome 21. This would be a female under the same circumstance. She has 45 chromosomes. She's a female because she has lost chromosome 14, lost chromosome 21, but gained the translocated Q arm of chromosome 14 and the Q arm of chromosome 21 here. Okay, so so you still have 2 copies of the genes, no problem in that individual here. Okay, but now again, what can happen though is in gametogenesis. Suppose this is going on in the germline. Okay. Now this is going to be the problem coming up. Suppose now in gametogenesis, what's going to happen is, now look at these 2 chromosomes here. So suppose in gametogenesis, this body thinks this is chromosome 14, and this is also chromosome 14. Even though it has a Q arm here of chromosome 21, the body thinks this is chromosome 14, and this is another chromosome 14 here. And so what can happen is, and this is chromosome 21, well, which it is chromosome 21. So what can happen is, in the offspring of this individual, so this chromosome 14 can go into one gamete, and this chromosome 14 can go into another gamete. And in that same gamete comes the normal chromosome 21. Okay, so there it is. So again, in half the sperm or eggs, if this is happening in the germline, what can happen here is and then that's under that circumstance, is that the cell in gametogenesis, it thinks this is chromosome 14, even though it has extra DNA coming from chromosome 14. So this is chromosome 14, it thinks so, and this is a normal chromosome 21. Everybody with me so far? Everybody with me? Everybody with me? Okay? Okay, all right. Okay, now, so this is in a gamete. So then it fertilizes the other gamete, And then in the other gamete is the normal chromosomes. Remember, haploid 23 chromosomes that are normal in the other gamete. And in the other gamete, there is a normal chromosome 21 and a normal chromosome 14. Notice what's going to happen in the zygote. What's going to, as well as the body, as the fetus being developed here. Notice what's going to happen. They're going to have two chromosome 14s. So they're going to be diploid for the genes on chromosome 14, no problem. But notice the genes in the Q arm of chromosome 21. We're going to have 3 copies of those genes on chromosome 21. 3 copies of the genes. Here it comes. Here it comes. What's the name of the pathology? What's the best example of Robertsonian translocation? There we go, Shireen. That's right. That's right, Vincent. Everybody else, that's right. That is Down syndrome. So Down syndrome, this is where they'll ding you if you're not paying attention. You better pay attention to this. This is extremely high yield. So Down syndrome can be caused for several different reasons. By far, the most common cause of Down syndrome is due to non-disjunction. 95% of Downs is due to non-disjunction, where it's a numerical alteration of the chromosomes, 47 chromosomes, because of a trisomy 21, extra chromosome 21. That was discussed earlier. Now, 4% of the Down syndrome patients have the Robertsonian translocation, where the total number of chromosomes are still 46, yet they have Down syndrome, and it's due to a structural alteration of the chromosomes. Natalie, I think that's what you were referring to earlier, but it is a structural alteration of the chromosomes, not a numerical alteration of the chromosomes, because they have 3 copies of the genes on the Q arm of chromosome 21. Here, this is Robertsonian translocation. Now, the other 1%, I never hear about that being on boards, but it's due to mosaicism. I wouldn't worry about that issue. But you got to know Robertsonian translocation because it illustrates this issue of what's going to go on in Down syndrome. They often ask this thing about Down syndrome. What can cause Down syndrome? And the total number of chromosomes is still 46. And you say, well, that's got to be Robertsonian translocation. Not reciprocal translocation. Not reciprocal translocation. This is Robertsonian translocation because it's the acrocentric chromosomes. You can lose the P arms. Again, you got to know the acrocentric chromosomes here. Okay, so Gisela, in one gamete, here is chromosome 14 and here's chromosome 21. Again, the cell is thinking this is chromosome 14. Remember, in that gamete, you're going to have 23 chromosomes. Chromosome 1, 2, 3, 4, 5, 6, all the way to chromosome 22. You have 22 chromosomes, autosomal chromosomes in that gamete, as well as one of the sex chromosome. Normal situation. But in that gamete, if Robertsonian translocation occurred, this is the chromosome 14 that got into that gamete. It's abnormal, but the cell thinks it's chromosome 14. Here is a normal chromosome 21. All the other chromosomes are normal, but that's in one gamete. Has an abnormal chromosome 14. Now, if it gets fertilized by sperm, or what if that was an egg and this is now sperm coming in with all 23 chromosomes that are normal, then we're going to get this situation, exactly what you see here. Here's the chromosome 21, 14 that are normal. Now you got 3 copies of the genes in chromosome 21. Okay, Gisela? Yeah, no. This is coming from one parent on the left side. This is coming from another parent, right? Okay, you see it? Okay, great. All right, good. I'm glad. You must understand this high yield stuff. Okay, does this Downs with this translocation have milder symptoms? I think they, yes, they do. I would ask Dr. Barone about that because I would be a little nervous to, I think they do, but I'm not 100% certain. But I would ask Dr. Barone later on. He's certainly going to be talking about Down syndrome. And you can ask him at that point there. I think they do, milder. Still tragic, but it is milder, I think. But I'm not 100% certain on that. So Dr. Barone certainly will know. Okay, all right, very good. All right, so now what's coming up next is, so this is the main reason why you need to know Robertsonium is because of the Downs situation. Okay, the next thing here is page 360 is another high yield figure, and it has to do with deletions. And this deletion we're going to talk about here is called a microdeletion, where a region of a chromosome is deleted. You got a big chromosome, and a region of it is deleted. That can be seen, there's such a big piece that's deleted, it can be seen in a microscope. These microdeletions. Okay, and now the one we're going to talk about here is in chromosome 5. And in chromosome 5, you must know because here is chromosome 5, the P arm and the Q arm. And now what can happen is in the P arm of chromosome 5, if you can have a terminal deletion, a region of the P arm is just on the terminal side, is just deleted, or we can have inside chromosome 5, a region can be deleted here. And that's called interstitial deletion, if a region inside the P arm is deleted. So interstitial versus terminal here. And now if you have a microdeletion of the P arm of chromosome 5, every medical student should know that gives rise to this pathology called Cri-du-Chat. You can either have an interstitial or terminal deletion of the P arm of chromosome 5, giving rise to Cri-du-Chat. And Cri-du-Chat, again, most students will get this. They have that characteristic high-pitched cat-like cry. They also have mental retardation, microcephaly, and so on. But the characteristic, you see this phrase on boards, high-pitched cat-like cry, you automatically say they're asking me about Cri-du-Chat. Now let me ask you this. What's that telling you about the genes on the P arm of chromosome 5? What do you think those genes are involved in? If you're deleting those genes and you get Cri-du-Chat, what pathway, what process what do you think they're involved in that results in the pathology? That's right. These genes are involved in larynx development, right? Yeah, that just makes sense. Here it comes. It makes sense, right? So you remove the P arm, a region of the P arm, and it gives rise. Then the genes that are involved in larynx development are not being made into enzymes. Larynx doesn't develop. The high-pitched cat-like cry. All right, now what I want to do here, again, Dr. Barone will go into more details of the pathology here. But now, what I want to do here, though, is to put this into perspective. And what I mean by this is these deletions. You could have, there's always something going wrong with chromosomes and genes and things like that. Always something going wrong. And so you can have big deletions, you can have small deletions, and so on. What I want to do is put things into perspective, try to help you out. And so we're going to start at the gross level all the way down to the smallest level of deletions. So what I want to do is at the gross level, the grossest level is where you can have, in terms of, by the way, other examples of diseases that are due to microdeletions is Williams syndrome. There's another example. But what I want to do is talk about at the gross level, is where you have a whole chromosome. Start at the gross level, work our way down to the smallest kind of deletion. So we're talking about deletions here. So the grossest type of deletion is a whole chromosome being deleted. And what I'm trying to get you to do is state what disease is associated with that type of deletion. Here it is. What disease is associated with a whole chromosome being deleted? That's right. There's only one pathology. We talked about it. As long as you were paying attention, you got this one. That's Turner's. Only one monosomy that's around, right? That's capable of surviving. Okay, now starting with the whole chromosome, if I ask you about a whole chromosome being deleted, you say, well, that's got to be Turner syndrome. Now moving down the ladder of complexity. Suppose we have a chromosome and a region was deleted, a microdeletion. There it is. You can say that's Cri-du-Chat is an example of a microdeletion. Williams syndrome, another example of a microdeletion here. Another example, we talked about it earlier, alpha thalassemia. Remember, a region of a chromosome because of unequal crossover can be missing. So alpha thalassemia also is an example of a micro deletion. A region of a chromosome is deleted. Now moving our way down in complexity. Whole chromosome can be deleted. A region of the chromosome can be deleted. What about a single gene being deleted? Let me see your answers here. What pathology is associated with a single gene deletion? Not hard. That's right. That's right. That is Prader-Willi and Angelman syndrome on chromosome 15. Let me ask you this: is Prader-Willi and Angelman, are they due to terminal deletion or interstitial deletion? Terminal deletion or interstitial deletion? Terminal or interstitial? No, that's right. It's interstitial deletion. It's in the Q arm of chromosome 15. It's not the tip of the chromosome 15. It's inside chromosome 15 where the gene is being deleted. All right, now moving down the ladder of complexity. Whole chromosome can be deleted. A portion of the chromosome can be deleted. A single gene in the chromosome can be deleted. And within a gene, again, it's interstitial deletion, but within a gene, a codon can be deleted. Here it comes from in pathology. What's the best example of a disease due to a codon deletion? Hector, not sickle cell. There we go. Cystic fibrosis. Cystic fibrosis. Well done, everybody. Cystic fibrosis. It's remember the deletion of a phenylalanine at position 508, delta F 508. Now, those of you who said beta thalassemia or sickle cell anemia, that's a missense mutation. There's no deletion. There's no deletion in sickle cell. There's no deletion in beta thalassemia. It's a one nucleotide change, not a deletion. Again, I want you to know this, that sickle cell is a change of a nucleotide. Here is a codon is being deleted here. All right. Okay. Now, what's coming up next is even beta thalassemia. There's a missense mutation in intron 1. Okay, and the gene is there. The gene is there is a one nucleotide change. The RNA that's made is not spliced properly, right? Okay, but the gene has no deletion. It has a change of a nucleotide. Okay, now alpha thalassemia, that's an example of a microdeletion. A region of the chromosome is deleted, alpha thalassemia. All right, now the simplest type of deletion is a single nucleotide deletion. Here it comes from pathology. What's the best example of a single nucleotide deletion? Single nucleotide deletion. No, that's change. Lucas, that's a change. Sickle cell is a change of an amino acid. There is change of a nucleotide. Remember, let me do it again. In sickle cell, in the beta globin, at position 6, there is a change, a missense mutation of an A replaced by a T. There's no deletion. It's just an A replaced by a T. The codon for glutamate goes to the codon for valine. But there's no nucleotide deletion. So sickle cell is not a deletion defect. That's right. Sukwant, see that question mark? Giselle, I see the question mark you have after Duchenne's. Delete, since we're talking about deletion, press the delete button for the question mark because you got it right. It is Duchenne's. That's right. Duchenne's is a one nucleotide deletion. And by deleting that nucleotide, it causes the frame to shift. So remember all that? The severe muscular dystrophy, that's Duchenne. That is Duchenne's. So look at this page. Look at all the pathologies here. How they could ask on the national board. That figure we had earlier in molecular biology about the different kinds of mutations in chapter 4. If you weren't understanding what I'm just saying here about sickle cell and so on, go back to that mutation figure. It is high yield. You see the high yield right here. We talked about it, integration of that molecular biology with genetics here. All right, extremely high yield. Now, Becker is an in-frame mutation. It's a missense mutation. Change. There's no deletion in Becker's here. Okay, now what's coming up next is, this is a high yield page. Now what's coming up next is the isochromosome. High yield. All right, now what's the isochromosome? Now, first of all, what is the definition of an isochromosome? An isochromosome is where you have two copies of one arm of a chromosome and no copy of the other. Okay, and normally, and usually I should say, usually it's lethal. But now let me tell you what happens here. Suppose we have a chromosome here. This is the P arm and here's the Q arm. And this chromosome is sitting in a cell in G1 phase. And then now the signal comes by a hormone to tell the cell go into S phase and begin multiplying. So sure enough, at the beginning of S phase, this chromosome looks like this. At the end of S phase, it now looks like this, right? The sister chromatid. Nothing strange. And then in M phase, what should happen to this sister chromatid? It should split right down the middle, right? That's disjunction. It should split right down the middle. And then as a result, this chromosome goes into one gamete, and this chromosome goes into another gamete. Nothing strange or unusual about this. But as I told you earlier, there's always something goes wrong in biochemistry. Instead of splitting right down the middle, what can happen is it can split horizontally. And when it splits horizontally, then we get this situation, where now the products, where by splitting horizontally, then as a result, we get fusion of the P arms and the fusion of the Q arms. And then they can go into respective into gametes and so on. And usually and in all cases when this happens, it's lethal. It's lethal because you're missing genes. I can imagine how devastating this is to the cell. It's lethal. The only situation where it can give rise to a person still living, which is compatible with life, is when this happens in the sex chromosome, specifically the X chromosome. With the autosomes, if this happens, it's lethal. But with the X chromosome, if this happens, then it is compatible with life. So when the Q arms fuse or when the P arms fuse, that's called the isochromosome. When the fusion of one arm but no copy of the other. So here we have an X chromosome. Suppose we have two of them, and in gametogenesis, what can happen is, or when the cell goes into S phase and starts multiplying, then as a result, what can happen is that we can form the isochromosome, where the Q arms now can fuse with each other to form the isochromosome. So then, as a result, then that will go into one gamete. It gets fertilized, and we end up with this particular karyotype. 46, X, and the I stands for isochromosome of the two Q arms of the X chromosome. Now you might say, where did the other X chromosome come from? Again, it's just meiosis. What I'm saying is this X chromosome, this isochromosome can go into one gamete, and then then it gets fertilized by another gamete here, which is normal. So this is now the normal X chromosome. This capital X right here is the normal X chromosome that's coming from the other parent. That's normal. And this isochromosome is coming from the other parent that has the isochromosome. And so as a result, we form this karyotype. 46 chromosomes, the total number of chromosomes is still 46. They have one normal X chromosome and one isochromosome. And why that's important to know is this gives rise to Turner syndrome. 22% of all Turner syndrome have the isochromosome. And why is that? Because the critical genes responsible for the Turner's phenotype are on the P arm of the X chromosome. But they're going to be missing the P arm. And as a result, they end up with the Turner's phenotype here. So Turner's syndrome, notice the karyotype. They have 46, the number of chromosomes, 46. Let me ask you this. So you might see Turner's syndrome on boards because of the issue of the isochromosome. What's another reason? What's the most common? If 22% of Turners is the isochromosome, what's the 78% that remains? What's that due to? What's that due to? That's right. That's the monosomy situation. That's the monosomy situation. So 78% of Turners is due to the monosomy. So that's another reason why you might see Turners. So one more time. You might see Turner's situation for a number of reasons besides the pathology. One reason is because it's the only monosomy in which a person can survive. You might see it on the boards because of the issue of the isochromosome. A structural alteration of the chromosomes, not a numerical alteration of the chromosome. This is a lot like Down syndrome. Down syndrome, it's most often due to a numerical alteration of the chromosomes, not structural. But you can end up with Downs with a structural alteration. Same thing here. Turner syndrome most often is numerical, but sometimes it's structural alteration of the chromosomes. It's basically the same concept here. Now, what's the other reason why you might see Turners on the national board? And the reason is because of mosaicism. Mosaicism is the 45X as well as the 46XX. Now, if they ask you what's causing mosaicism in Turner syndrome, and the answer is it's because of non-disjunction in mitosis, not meiosis, mitosis. In other words, after the embryo forms, after fertilization, and then you form the blastocyst, the embryonic stem cells. What can happen is in some of those cells, they can lose the X chromosome, and so therefore some of the cells will differentiate with a normal karyotype, 46XX, and some of the cells, because they lost the X chromosome, can differentiate into cells having the 45X phenotype, genotype, karyotype there. All right, so again, the major issue of the Turner syndrome here. You got to know Turner's. Now, again, all the pathology Dr. Barone will talk about. Most often on board exams, they'll give you a vignette about a patient who has all the characteristics of Turner's. So you got to know the pathology. They don't say Turner's. They say it gives you all the characteristics. Then they ask you something about the genetic issue. That's how they'll do it on board exams. Now, one last item. There's things called ring chromosomes and so on. Paracentric inversions, low yield. We're going to skip over that. There's one more item I want to talk about, and that's called the FISH assay. Now, what the FISH assay is, very simple. If you were asked, what's the FISH assay about? Fluorescent in situ hybridization. In this chapter, as well as chapter 1, we talked a lot about how this particular gene is located on chromosome 7, this gene is located on chromosome 15, and this kind of stuff. How do we know which chromosome a gene is located? So the idea is, so I can go and make a fluorescent probe, against that's going to be complementary to a particular gene. So where do you get the fluorescent probe? I just call a company, and say, send me the fluorescent probe for insulin, because I want to know which chromosome the insulin gene is located. So now in my hands, eventually I'll have the fluorescent probe against the insulin gene. So then I take a biopsy of me or whatever and lay out the chromosomes. It's going to locate genes on a particular chromosome. So I lay out the chromosomes on a glass slide, and they're laid out, numerical order, whatever, and then I add the fluorescent probe. The fluorescent probe will now look at all the chromosomal DNA, look in the hybridized, look in the base pair, and suppose it finds the insulin gene on this particular chromosome right here, on the P arm of this particular chromosome. If it happens to be chromosome 1, 2, 3, 4, this is chromosome 4, say, well, the insulin gene is located on the P arm of chromosome 4. That's what it means, FISH assay. It's fluorescent in situ hybridization, where you take a fluorescent probe, not a radioactive probe, a fluorescent probe used to locate a gene on a particular chromosome. In case you see that issue on board exams, or where they might say to you, a FISH assay was used to do something or whatever, that's what they're referring to. Now, what I want to do is to conclude this chapter with one vignette. And this is a very difficult vignette. So if Carrie could set the timer for 78 seconds. This is a When I do this vignette for students, they generally, a lot of students get it wrong. That's why I'm doing it. And it's at the end of the chapter, so again, don't look at the answer because you're not fooling anybody. Try it out on your own first. Okay, so here comes the vignette. Ten seconds. Okay, and we're going to close the poll and broadcast the results. And you can see that by the time, you don't have a whole lot of time, 78 seconds go by very, very fast. That's why you got to think quick. Okay, and it looks like over half of the students put option E, and the answer to this question is option D here. All right, so we got some, obviously we got some explaining to do. You can see why I'm doing the question, because I know most students get it wrong. So only 18% of the students got it right here. Okay, so let's go through it. Why option, now before we talk about why option D is correct, let's go through why the other options are incorrect here. Why they're incorrect. Option A. So here is now here's the mother's whatever chromosome numbers these are. These are two chromosomes of this particular chromosome number, and here's two chromosomes of that chromosome number in the mother. Here's the father, and here is the fetal tissue. Now it says over here, aneuploidy in the fetus. What's telling you, what's wrong? Why is option A incorrect? Aneuploidy in the fetus. Why is option A incorrect? Let me see your answer. Why is option A incorrect? Let me see your answers. That's right, because aneuploidy means a numerical problem, right? Aneuploidy means a numerical problem. All the other chromosomes are around, so don't worry about the other chromosomes. But notice here, this is a structural alteration of the chromosome, right? It's a structural, by color coding, the tip of this chromosome here is now found over here, right? So that's what this is, a structural alteration, not a numerical alteration. So that's why option A is incorrect, because the fetus is euploid. The fetus has 46 chromosomes. If it was aneuploidy, it would be not 46. So all the other chromosomes are not around, but they're there. I mean, just not drawn for you. So the fetus is euploid. So it's not aneuploidy. Option B, fetus is identified as the reciprocal translocation carrier. Why is that incorrect? Why is option B incorrect? Fetus is identified as the reciprocal translocation carrier. And the answer is, it's not the fetus that's the reciprocal translocation carrier. It's the father. The father is the reciprocal translocation carrier. Notice here, if the mother has the normal chromosomes, here is these two chromosomes are normal, these two chromosomes are normal. But what happens is in the father, the P arm of this chromosome now comes onto this chromosome, and the P arm of this chromosome goes onto that chromosome, as shown by the color coding. See that dark blue there is now found over here. There it is, right here. And that light part there is now found over here. Okay, right there. There it is. So again, the father there's no loss or gain of genetic information in the father, just the tips of the chromosomes are found on different chromosomes. So the father is a reciprocal translocation carrier. He's alive, he's fine. It's the fetus that's going to suffer. Okay, now now option C, non-disjunction during oogenesis in the mother. Why is option C incorrect? Non-disjunction during oogenesis in the mother. Why is that incorrect statement? Non-disjunction means numerical alteration of the chromosomes, right? Okay, again, by the way, I'm just pointing out here, this is the reciprocal translocation that took place here. Okay, now option C. The non-disjunction during oogenesis, they would have the normal chromosome structure, right? Non-disjunction means nothing wrong with the structure. It's the numbers game. Nothing wrong with the structure. Clearly, this is a structural problem in the fetus. Now, most students put option E, and that's wrong. Unbalanced chromosomal material in the father. Why is that incorrect answer? Because the father is the reciprocal translocation carrier. He would have a balanced number of chromosomes. It's just whatever genes are here and here, they're in different locations, but the father is still diploid. That's right. Nothing's being deleted. That's right, Mithra. That's right. Now, option D. Why is that the correct answer? Partial monosomy and partial trisomy in the fetus. Where is the partial trisomy? And the partial trisomy is right there. Notice whatever genes are located on the tip of that chromosome, there are 3 copies of it. There is the partial trisomy. And where is the partial monosomy? It's right there. So whatever genes are located here, you only have 1 copy of those genes. And the cell can't survive the partial trisomy and monosomy in the fetus. That's why the fetus is miscarriage. There it is. Okay, so what we're going to do is we're going to take a 10-minute break, and then we're going to come back, and then we're going to get into chapter 4. Very simple, only 1 figure in chapter 4, and 3 figures in chapter 5, and then we'll get into chapter 6. So we'll be close to finishing genetics tonight. I'm going to try to get genetics done tonight. So anyway, we're almost right where I want to be. Let's take a 10-minute break. See you shortly. I see in the public chat pod that Iman says, why will the father have no disease in this example? Remember, he's a reciprocal translocation carrier. Notice here, there's no loss, there's no gain of genetic material. There's no loss of genes, there's no gain of genes. The location of the genes are different, but there's no loss. That's why there's no pathology. He's not losing any genes. He's not gaining any genes. So that's why there's no pathology in the father. But he's a reciprocal because he underwent reciprocal translocation. But he has no pathology. As we talked about earlier in the chapter, the disease pathology can arise, or lethality can arise in the offspring of that individual. That's why. Okay? There you go. All right, now what's coming up next is chapter 4. Now, chapter 4 is, now this is why, again, I would not advise reading ahead, because in chapter 4, it's a low-yield chapter, genetics of common diseases. In this, there's only one figure I'm going to show, and then we're going to be done. And that figure is on the definition of multifactorial inheritance. What does that mean? Multifactorial inheritance. What does it mean? The rest of the chapter dealing with concordant twins and this and that, who cares? Don't worry about the rest of that stuff. There's only one item you need to know that that might be medium yield, and that's multifactorial inheritance. Everything else, forget about. So this is a low yield chapter. One figure only. Why waste your time, right? You don't want me to waste your time on stuff that's unlikely to be on board exams. Now, what does multifactorial inheritance mean? What that means is most common diseases are caused not by one gene defect. Chapter 1, that was single gene diseases, single gene defects that cause disease, like PKU, sickle cell. One gene caused a particular disease. But most common diseases are caused by multi-factors. Look at the words, multi-factors. Multi-factors like what? Like defective genes, multiple gene problems, and environmental factors. Now, an example of a common disease that displays multifactorial inheritance is the disease called it's obesity. Obesity. Now, what I mean by this, and I'm not talking about being overweight. I'm talking about severe obesity. Okay. Now, the idea here, the concept here is what I want to do is a theoretical situation. The theoretical. Suppose one gene was responsible for the BMI, body mass index. BMI, body mass index. Suppose one gene was responsible for the BMI. So what that would mean is everybody listening to me in the audience right now, everybody would we would all have the same BMI, and suppose that BMI would be a single BMI, and suppose that BMI body mass index was set at 26. So everybody, if one gene was responsible for the BMI, everybody listening to me right now would have a body mass index of 26. But as you know, any gene can be mutated. So if the gene got mutated, then there will be two BMI values. One which would be normal of 26, and the other one, let's say, that would be 38. So in other words, most of the people listening to me right now would have a BMI of 26. And if in some people that gene got a missense mutation or nonsense mutation, something like that, then the gene got mutated, and then the BMI would be 38. And then because we are diploid, what about the heterozygous situation, where we would have one normal and one mutant gene, and then you would have three BMIs to have the heterozygous situation. And let's say that the BMI, therefore, we can set it at 32. So there would be three BMI values. Most of the people listening would have a BMI of 26. Some lesser people would have a BMI of 32, and a much less number of students would have a BMI of 36. If one gene was responsible for the BMI. Okay. But as everybody knows, that's not what really happens in obesity, because obesity is caused by a great number of multiple gene problems and environmental problems. So what happens is nutritional experts and so on, they get together and they decided to set the threshold of obesity, of the clinical problem of obesity, at about 30 kilograms per meter squared. So at 30 kilograms per meter squared or less, a person has a normal BMI. Depends on that particular person and the height and all that kind of stuff. Any value that a person has of a BMI greater than 30 kilograms per meter squared will be much more labile. Press the wrong button. Much more labile to have a very severe clinical problems here. Like now, what kind of genes would cause would cause obesity? Well, as everybody knows, if there's problems with eating too much carbohydrate, the genes that are going to be affected, there'll be the fatty acid synthesis genes. Too many genes are being turned on, and you're going to be making a lot of fat here. Other things can go wrong, environmental issues. What kind of environmental issues? Like going to McDonald's all the time. If you go to McDonald's breakfast, lunch, and dinner for a whole month at a time, you're going to gain weight, right? That'll cause your BMI to go to the right. So environmental problems, gene issues will cause the curve to go to the right. So that's called multifactorial inheritance. They made a movie. Where a couple years ago, went to the McDonald's for a whole month, breakfast, lunch, and dinner. What was the name of that movie again? The name of the movie where the guy went to McDonald's. That's right. Mithra and Lucas. That's right. Super Size Me. That movie was about biochemistry here. That movie was demonstrating what I'm just showing you on page 372. They should have called the movie page 372, because that's what the movie was all about. Okay, that's right. Now what's coming up next is, so that now we're done with the chapter. There it is. Simple. Make it simple. Don't overdo it. Don't bore the students. Don't waste your time. I don't want to waste your time. So now the rest of the chapter, concordance, forget about it. Now what's coming up next is chapter 5. And it is, I would view this as a medium yield chapter. And unfortunately, a lot of students don't like what's coming up here. Because what happens is there's going to be some mathematics. And the concepts that deal with the mathematics, some students have a hard time with. So I will try to keep it simple. I will try to make it understandable, because we got to talk about recombination frequencies and things like that. And I know a lot of students just get a mental block against this stuff. But I'll try to take it and be as simple as I can in explaining what goes on here. But because it's a medium yield item, at the very end, if you still don't understand it, don't panic, because it's medium yield. You might see it, you might not, you might, take a chance. If it's low yield, definitely take a chance it won't be on boards. But if it's medium yield, well, you still don't waste too much time trying to understand it. Focus your attention on the high yield items. So in this chapter, there are 3 figures that we need to talk about. Then we'll move on to chapter 6. So the first figure to talk about is what's recombination frequency about, and this issue of linkage. Okay, so what this is about is in this figure, you're going to, as well as 2 other figures coming up, you're going to be seeing letters here, G and M. G stands for some gene, and the M stands for a marker. Remember the other day we talked about those STRPs that can be either STRs. Remember, they can be either 2 to 6 nucleotides in length. Remember all that kind of stuff? So we talked about markers earlier. So in this chapter, please don't think that the marker causes pathology. The marker does not cause pathology, but it can be used for genetic counseling, as we will see in this chapter as well as in Chapter 6. So the marker can be used for genetic counseling. Okay, so now the issue of linkage. Linkage means that the alleles of two loci are likely to be inherited together. What does that mean? What that means is, suppose we have a cell in somebody's germline that getting ready to go undergo meiosis. So suppose we have a pair of chromosomes here with a gene, and suppose we have two versions of the gene. Gene 1, which may be normal, and gene 2, which may be mutated. And then on another chromosome, we have that marker. We have two versions of the marker, marker 1, marker 2. So what I mean by that is, whatever the marker is, whatever the STRP is, here it might be amplified 4 times in a row, and the other chromosome, that marker might be amplified 6 times in a row. 2 different versions of the marker on other chromosomes. So again, the concept is, so we have 2 loci, two locations. One location has a gene, and the other location has a marker. And there are 2 alleles of the marker and 2 different versions and 2 alleles of the gene. Nothing strange. Now again, if it says if the gene and the marker are on different chromosomes, if the gene and the marker are on different chromosomes, then in the process of meiosis, what's going to happen is that if a cell picks up the chromosome that has gene 1, 50% of the time it's going to pick up the chromosome that has marker 1, and 50% of the time it'll pick up the chromosome that has marker 2. So one more time, it spells it out here. If a cell gets gene 1, then 50% of the time it's going to pick up the chromosome that has marker 1, and 50% of the time it'll pick up marker 2. And then therefore that means that gene and the marker are unlinked. Unlinked. They have nothing to do with each other, and the marker cannot be used for genetic counseling, as we'll talk about a little bit later. All right. Everybody with me so far? That's right. Pay attention, because I know what's going to happen is, if you're not paying attention, you're going to go, I don't understand what you said. Then you're wasting time. Okay. So far, so good. Everybody with me? Okay. Everybody with me so far? That's right, Mithra. That's right, Mithra. You tell everybody. Mithra, be my right-hand person. When people are not paying attention, you spank them. That's right. You tell them pay attention. That's right. Okay, well done. Okay. Now suppose, because this gets complicated if you don't understand this initial stuff here. Okay, now what happens now, suppose the gene and the marker are far apart on the same chromosome. Okay, so here it is. Here we have one chromosome here, we have and the homologous chromosome, there it is, and here we have the gene 1, gene 2, and on the same chromosome we have marker 1 and marker 2. Now remember, if they are far apart on the same chromosome, because of the issue of crossing over, that's translocation. Remember, that's normal. We talked about that in chapter 3. Crossing over between homologous chromosomes are normal. That's what you're trying. Think that right now. Nothing strange, nothing unusual, no pathology. Crossing over between homologous chromosomes. So now, because of crossing over that can take place, because if the gene and the marker are very far apart on the same chromosome, then because of crossing over, that if a gamete in the process of meiosis, if a gamete gets gene 1, it is 50% of the time it'll pick up marker 1, and 50% of the time it'll pick up marker 2, and that's all normal because of crossing over that occurs. And then that would mean that, again, one more time, if they are far apart on the same chromosome, if a cell gets gene 1, 50% of the time it's going to pick up marker 1, and because of crossing over, then 50% of the time it picks up marker 2. Then therefore that means the gene and the marker are unlinked. And again, they have nothing to do with each other, and the marker cannot be used for genetic counseling. So now there's another definition that we need to attend to here. So again, this is the genotype of the gene, gene 1, gene 2, and this is the genotype of the marker, marker 1, marker 2. But there's another term called the haplotype, shown on page 388 of the book. A haplotype, by definition, is the combination of alleles on the single chromosome. So the haplotype here would be gene 1, marker 1. In the other gamete, the haplotype is gene 1, marker 2. So one more time. If the gene and the marker are very far apart on the same chromosome, if a cell gets gene 1, 50% of the time it's going to pick up marker 1, and 50% of the time it'll have marker 2 associated with it. And the gene and the marker, get ready with a highlighter, are unlinked. Nothing to do with each other. But now the bottom panel. If the gene and the marker are very close together on the same chromosome, here they are, in the same chromosomes, here's the haplotype, gene 1, marker 1, very close together. On the other chromosome, gene 2, marker 2. Then, in the process of meiosis, if a gamete picks up the chromosome that has gene 1, it is likely to still have the marker 1 associated with it, and very little probability of having marker 2, because crossing over is going on, but the closer the gene and the marker are to each other, then they're likely to be linked. So again, if a cell picks up, if a gamete picks up gene 1, it is likely to pick up marker 1 also. That means the gene and the marker are linked. So this gives rise to the term recombination frequency. Now this, again, the recombination frequency is about, it's the closer to 0% the recombination frequency is, the closer the marker is to the gene. If I say to you, the recombination frequency of the marker and the gene are 40%, that means, well, they're on the same chromosome, but they're not too close together. If I say to you the marker and the gene has a recombination frequency of 10%, that means the marker and the gene are pretty close together. If I say to you that the marker and the gene have a recombination frequency of 1%, that means the gene and the marker are very, very close together. If I say to you the gene and the marker have a recombination frequency of 0%, that means the gene and the marker are side by side on that same chromosome. So the closer to 0% the recombination frequency is, the closer the marker is to the gene. Okay? Everybody with me so far? Okay. All right. Now what's coming up next is this is going to be a little complicated now. This is how the marker can be used if the marker is close to the gene in terms of genetic counseling. Okay, now what's this about is we're going to talk about neurofibromatosis type 1 here, and the gene is on a particular chromosome, chromosome 17. So the idea, don't worry about the chromosome number here. Now suppose on chromosome 17, as shown in the orange, that's the neurofibromatosis gene sitting on chromosome 17. So we have 2 copies of that gene, and remember, the gene can be normal or the gene can be mutated. And let me ask you this quickly. What mode of inheritance is neurofibromatosis type 1? What mode of inheritance is it? Everybody should know this. That's right. It's autosomal dominant. That's right. So we can have 2 different versions of the gene. And let's assume the person has capital A on one chromosome and little a on the other chromosome. And remember, because it's dominant, capital A is the mutant allele, the Z allele, and little a is the normal allele. So the genotype could be little a. The genotype could be capital A, little a, like you see right here, or the genotype could be capital A, capital A. Okay, so that's the genotype. Now suppose nearby the gene there is a marker, 2 different versions of the marker, shown here in the green. 2 different versions of the marker, and we'll call it marker 1 and marker 2. Again, what do I mean by marker? It's a STIRP. In this case over here, the marker might be amplified 5 times, and over here, the marker might be amplified 10 times. 2 different versions of the marker. It's not causing pathology, but it's near the gene. Okay, now what I'm going to do over here in this pedigree right here, what I'm going to do is each of these individuals, we're going to talk about the haplotype, genotype. Now, what I mean by that is, see these numbers underneath all of the people. Now, remember earlier, if you see numbers in a pedigree underneath people who have the disease only, that has to do with those triplet expansion repeat diseases, the age of the individual and the number of repeats. But here you see numbers that have in all of the individuals. So if you see this on the national board, what's that referring to is the genotype of the marker. So the genotype of the marker here is 2-2, the genotype of the marker there, and this female is 1-2. Now, what I'm going to do over here is I'm going to go each individual. We're going to talk about the haplotype, genotype of each of the individuals. Let's start now with this female right here. This female, the pedigree says, has the disease, and it says the marker is 1-2. So what is the haplotype, genotype of this individual? In other words, what do her chromosome 17s look like? She has 2 chromosome 17s. What do they look like with respect to the marker and the gene? And the answer is she would have this genotype, haplotype. One of her chromosomes would have capital A1, and the other chromosome would have little a2. Exactly what you see right here. In other words, this female is this these 2 chromosomes that you see right here. Okay. Because it says the female is affected, so she has to have a capital A, and it says the genotype of the marker is 1-2. Now the male that she mates with is genetically unrelated, and therefore his genotype and haplotype got to be little a2, little a2. He doesn't have pathology, so both of his neurofibromatosis genes are little a, little a. This says the genotype of the marker is 2-2. So nothing strange, nothing unusual. Everybody with me? Everybody with me so far? Okay? Oh, yeah, good. Okay. All right, now, so then they mate, and then they have a female, a daughter, and who has the disease. So now, what did the daughter inherit from her father? Which chromosome 17 did the daughter inherit from her father? And which chromosome 17 did the daughter inherit from her mother? And it says, because she has a disease and because her genotype of the marker is 1-2. What is her haplotype, genotype? And the answer is, that's right. Her haplotype, genotype is capital A1, little a2. Now, how do you know that? Because from her mother, because she has a disease, from her mother, she has to inherit this particular chromosome right here. She's inheriting this capital A1 chromosome right there because she has a disease, and it says her marker is 1. The father doesn't have the 1 marker. And then from her father, she's inheriting the little a2 chromosome. Okay, so there it is. So notice here, so far, in this family, not outside the family. When you talk about markers, you talk about only in a pedigree, only in a family. Notice how the allele 1 is linked to the marker here. The allele 1 of the marker, I should say. The allele 1 of the marker is linked to the disease phenotype here. Okay, but now this female then mates with her husband, and his marker, he doesn't have the disease, and his marker genotype is 1-1. So even though he has 1-1, why doesn't he have the disease? And again, note that this person here has the 1-1 marker but doesn't have the disease. Why is that? No, nothing to do with incomplete penetrance. No, nothing to do with that. It's simply he has 2 versions of the marker. He has 2 normal little a's. And his marker is 1-1. Remember, he is genetically unrelated. This man is not genetically related to these 2 individuals. He is not genetically related to these individuals. He is, so again, to understand the markers, you got to talk about those people who are genetically related. This person is not genetically related to these 2 individuals right here. All right, so you got to understand, the pedigree says he doesn't have the disease. He's outside. He's little a, and the genotype of the marker, it says, is 1-1. Okay, so then these two mate. And what happens there is they end up having 6 children here. And some of them have the disease and some don't. Okay, now we're going to go. Now what happens is, what I'm going to do right here is the genotype haplotype of this first child here is capital A1, little a1. In other words, this son inherits the capital A1 chromosome from the mother and little a1 chromosome from the father. And because the capital A, the person has a disease. Now what I'm going to do here is I'm going to show this another way. What is happening? What is happening? To try to make it understandable. I could blow this by you, but I want you to understand this. Now suppose the mother, it says the mother, this mother right here, generation 2, she has a capital A1 chromosome and little a2 chromosome, like that, because that's what it says. Okay, so in meiosis, what's going to happen is, in half her eggs, she's going to have the capital A1 chromosome. Other eggs, she'll have the little a2 chromosome. Okay, in their eggs. And then she mates with this male, and his sperm. It turns out all of his sperm, let me do the color coding. So his sperm, the chromosome 17, will be in the yellow here. So all of his sperm chromosome 17s will have the little a1 haplotype, because it says so. Okay, so all of his sperm have this haplotype, little a1. So if this sperm fertilizes that egg, then the result will be capital A1, little a1, exactly what the son has. In other words, this son came about because of the fertilization of this egg, this sperm with that egg. And because of the capital A, that's why he has a disease. If this sperm fertilizes this egg, then the combination will be little a2, right here, the little a2, little a1 from the father, and this results in the daughter, right here. There it is. That's the daughter. Little a2 and little a1, right? Everybody with me? There it is. Just check the numbers underneath. There it is. So it's just, so in these first 5 individuals, the first 5, don't worry about this person here yet. We haven't got to that yet. In these first 5 individuals, everybody who has the marker genotype 1-1 has the disease. Everybody who has a genotype 1-2 will not have the disease. So if that's all there was, they can have 10 children, and every genotype of the marker of 1-1, they're going to have the disease. Everybody who has a genotype 1-2 will not have the disease, if that's all there was. Okay, everybody with me? Just by the beauty of the marker, if that's all there was. All right, but notice over here. Here is so all these people here, 1-1 have the disease, 1-2 do not. 1-1 have the disease, 1-2 does not. But now we come to child 6. This child has the 1-1 marker and doesn't have the disease. And because they do not have the disease, what's that due to is, that's due to, so again, there's no recombination took place in these individuals here. Why this person does not have the disease is because recombination took place. Recombination took place here in that individual. Now to explain what goes on, see these 2 chromosomes here? Suppose recombination took place, where the tips of the chromosomes in the female just underwent recombination. And so as a result, capital A is now associated with with marker 2, and little a gene now associates with marker 1, just because of recombination took place. And then in the process of meiosis, then we get this situation where some of our eggs will have the capital A2, and some of our eggs will have the little a1 chromosome 17. And then the sperm will have the little a1. The father has the little a1. There it is. There's a little a1 coming from the sperm having a little a1. If this sperm fertilizes that egg, we get this combination of the chromosomes, capital A2 having the disease, little a1. And if this sperm fertilizes this egg at the bottom, we get this combination, little a1, little a1. And because there's no capital A, that means no pathology, has not inherited the capital A. So therefore, and that is exactly what this son was due to. It was due to recombination that took place here. Recombination. Okay, one more time. Okay, one more time. So the idea that don't think of the father, think of the mother. The father has a little a1. They can undergo recombination, but it's always going to be little a1, little a1. They got 10,000 recombinations. It's always going to be a1 here. But it's in the mother. Here is the mother, because the recombination took place, or just the recombination took place. Now the capital A gene is associated with marker 2. Little a gene is associated with marker 1. And then in meiosis, this goes into one egg, that goes into another egg. If this sperm fertilizes that egg, we get this combination. If this sperm fertilizes that egg, we get this combination. So as a result, that's what this individual is right here. He is a recombinant. So the concept here is that now, of the 6 individuals, and again, you have to think only in this family, not outside, in this family, because we're looking at that particular marker. So in this family, so it says the gene is linked to the marker because they are inherited more than half the cases. Of the 6 children in generation 3, one of them is a recombinant, little a1 coming from the mother. There's a recombination that took place, the little a1 now coming from the mother. So of the 6 children, one of them is a recombinant. So then if the question becomes, what is the recombination frequency? Out of 6 individuals, 1 is a recombinant. What is the recombination frequency? It's 1 divided by 6, right? 1 divided by 6. So the recombination frequency is 1 out of 6, which turns out to be 0.17. Which turns out to be 17%. So the recombination frequency is 17% here. Okay, now that's right. Well done, everybody. So that's the 17%. So the idea is now and it turns out that's the distance between the 2 loci. Remember, I told you earlier, the closer to 0%, the closer the marker is to the gene. If the marker and the gene had a recombination frequency of 0%, they would be side by side, and recombination would not be occurring at that because they're side by side. The closer to 0%, the closer the marker is to the gene. But here it's 17%. So what happens is in genetics, there is a term that's called centimorgan. So for each 1%, it's equal to 1 centimorgan, cm, which turns out to be 1 million nucleotides. In other words, the distance between the marker and the gene is 17 million nucleotides. So the recombination frequency, again, if they say to you the recombination frequency is 1%, that means the marker is very close to the gene, a distance of 1 million nucleotides. If they say the recombination frequency is 0%, they're side by side. But in this example, the recombination frequency is 17% here. So they're not close together, but they're not far apart either here. Okay. Now let me ask you this. Do you think, so hopefully everybody's with me here, in this family, that's right, in this family only here. Okay. Now let me ask you this. Now think. Do you think that the actual distance between the marker and the gene is really 17 million nucleotides? Do you think that's really accurate number, 17 million nucleotides? No, it's not very accurate. Based on 6 children, I mean, if they have a 7th child or 8th child, I mean, so the idea is based on 6 children, it's not very accurate here. So we know that it's not very accurate. So what happens is, now this is the next part here it's amazing. It's can be very difficult, but it won't, because I want to make it simple. What happens is, because it's not very accurate, the recombination frequency based on one family is not very accurate. What happens is geneticists, statisticians, computer jocks get together and they take information from many families of a particular disease, many families, and they go through all kinds of algorithms and all these type of computer programs to come up with a little bit more accurate information. And that's what's coming up on the next figure, called LOD scores. Now when you see these numbers coming up on the next figure, when you see these numbers, don't ask, where did the numbers come from? How did you derive the numbers? Because that's all that computer program stuff. Don't worry about where the numbers come from. Just worry about how you interpret the numbers. Don't worry about where the numbers come from. Now, if you're a type A personality, you got to know everything. Like I want to know where those numbers come from. I'm telling you right now, if you try to worry about where the numbers come from, I'm telling you right now, blood is going to shoot out of your eyes, and your head is going to explode, right? Because if you want to know where those numbers are coming from. So don't worry about where they're coming from, okay? All right. So now the issue is, he got scared. It's not Halloween, but at least you're paying attention. At least you're paying attention. Okay, so again, the idea is it's just a cartoon. Don't worry, it's not a real person. It's not a real person. It's a cartoon animation. Okay. All right. Now the idea is now what this is about, again, these LOD scores. Again, LOD scores is data taken from many families here. Now the idea is the LOD is the probability of a gene and the marker are linked together. It's coming from many families. Now all you need to know, that how you interpret the information is very simple, and it's basically right here. This is the important feature. The idea is you first look at the LOD scores and ask, is there a number greater than 3? And in this example, there is. If there's a number greater than 3, it means linkage. And then all you got to do is go to the recombination frequency just above it, and that is the recombination frequency. So again, don't worry about if it's less than, or if it's, what about if it's 1 or 2 or something. Just on board exams, they're going to say greater than 3. So the idea is if there's an LOD score greater than 3, it means linkage. So you look at the values greater than 3, and then you look at the recombination frequency just above it, and in this example, it's 0.1. And so that is the recombination frequency, 0.1 or 10%. That is the probability. That is the recombination frequency of the linkage of the marker to the gene, 10%, coming from many families. Now, if there are two numbers greater than 3, then you look at the highest number greater than 3, and you look at that recombination frequency just above it. Now, as you all know, you get assessment exams online, and you will get assessment exams for biochemistry, as well as separate exam for genetics. It's a 20-question exam, and in that medical genetics assessment exams, there will be an LOD vignette, and it looks very difficult, but it's simple if you just paid attention to what I just said, how you interpret the results. It's just simple, and you will see, man, that's a pretty easy question. I hope they ask me that question on the board or one like it. It's very simple. So as long as you know this information right here. Okay, so now what's coming up next is, so now we're done with the chapter. Now we're done with it. There it is, all simple, and it wasn't that difficult. So again, this is the two loci are likely to be linked at a distance of 10 centimorgan, 10 million nucleotides from each other. Okay, so now what's coming up next is the last chapter. And the last chapter is somewhat related to stuff we talked about in molecular biology, is genetic testing, genetic diagnosing. Now the idea here is that if we have on a chromosome, if we have a gene that's mutated, we can, there's things called indirect or direct genetic diagnosis. In other words, we can directly look at the mutation. How can we directly look at the mutation? Where you look at the mutation itself. In other words, if in your clinic some family comes in with a particular trait, and you want to see who is homozygous, heterozygous, you can look at the mutation directly. And what kind of techniques can be used? Techniques like PCR, dot plots, that's coming up in the next figure, RFLP analysis. You can sequence the gene to see where the mutation is, that kind of stuff. You saw this earlier. Remember that one question in molecular biology that dealt with sickle cell anemia? Remember the family had a son and the fetus, and they want to know about the fetus was a carrier for the trait. Remember all that? Because we did RFLP, because we could directly look at the gene itself by a variety of techniques. But there's another thing called indirect diagnosing to do counseling of a family, is where you don't look at the gene directly. You look at a marker that's linked to the gene. And again, this is indirect diagnosis, where these linked markers that have to be linked, they have to be close by with some recombination frequency, and let's say 10%, very close by, are used to infer whether an individual in a family has inherited the disease-causing mutation. And how can you examine the marker? You can examine it by PCR, RFLP analysis, STIRP analysis, something along that line. Now, in this chapter, there are going to be 4 figures that we're going to go through. And 2 of them deal with how you directly look at a mutant gene, and 2 of them deal with how you look at the marker and not the gene. You will see the beauty of the marker. And the first one we're going to talk about is the use of a dot plot to directly look at a mutation, and the disease there will be hemochromatosis. Right after that, we'll talk about RFLP analysis to analyze a family with Gaucher's disease. After that, there'll be an example of looking at a family with Marfan syndrome by looking at a particular marker, and what's being analyzed there is a particular STIRP. And then lastly, we'll look at we'll do an RFLP analysis by looking at the marker that's linked to a gene that causes Lesch-9 syndrome. So the idea is this is what's going to be discussed in this chapter. Okay, so we got a little bit of time, so let's try to get this stuff done tonight. There's 4 figures, then we'll be done tonight. Okay, and then we're done with genetics. Okay, so now the first figure is the dot plot. Now the dot plot is, this is going to be the first time we talked about the dot plot. Now when we talk about the dot plot, we got to talk about things called ASOs. Now what is an ASO? ASO stands for allele-specific oligonucleotide. Allele-specific oligonucleotide. Okay, and what does it mean? It's specific to either a normal gene or a mutant gene. It's specific to whether it's normal or it's mutated. So ASOs. And when you hear the words ASOs, you always talk about 2 of them. You always use 2 ASOs. And they're usually very small, about 10 to 20 nucleotides in length. So very small ASO probes are used. And why you always need 2 ASOs? Because one which recognizes the normal allele, but not the mutant allele. One which recognizes the mutant allele, but not the normal allele. They're specific to what they're looking for. And they're very, very small, 10 to 20 nucleotides in length. Where does the ASOs come from? They come from Walmart. You go to Walmart over there, you will buy some ASOs. They're commercially available right now, and so on. Now in this example, we're going to talk about hemochromatosis. There is a gene called the HFE gene. When that gene is mutated, it gives rise to iron overload. You absorb too much iron into the body. Now, just like sickle cell anemia, where we know where the mutation is, same thing here with hemochromatosis. we know precisely what the mutation is. And we know the mutation is at position 282. So what you're looking at here, this is a portion of the gene for HFE, and the first 279 codons are not even shown here for the gene. This is position 280, 281, and 282. As shown by the arrow, there is a G at this position, and so on. Now, in hemochromatosis, it turns out there's a missense mutation, just like sickle cell. It's a missense mutation because we know what the mutation is at position 282, where this G is converted to an A, just by a missense mutation. So as a result, in the mutant gene, it's called C282Y. And what the C stands for is, this is the codon for cysteine, this is the codon for tyrosine. So at position 282, there is a replacement of cysteine to tyrosine. No one expects you to know that. Just know that it is a missense mutation. So now, since we know what the mutation is precisely, we can now design probes, these DNA probes, that one specific for the normal allele and one specific for the mutant allele. So now at the bottom of the figure here, notice here is the ASO that's designed to see the normal allele. It's the sequence right here. You buy it, or you can make it in the laboratory. And notice this is the 3' end. It says so. It's the 3' ATA all the way to the 5' end. And then we make another probe that's specific to the mutant allele. Okay, and so these two probes are identical to each other, except there's a C at this position and there's a T at that position in the mutant probe. Now watch what happens by PowerPoint. If I take the normal ASO that sees the normal allele, watch what happens. So if I take the normal probe, the normal ASO, and give it to the normal allele, here it comes. I'm going to give it to the normal allele. Watch, watch, watch. It's going to base pair. 100% base pairing, where all these nucleotides base pair with each other, and even the G base pairs with a C at that position 282. So 100% base pairing. So the normal probe sees the normal allele, but not the mutant allele. It will not base pair with the mutant allele. Why not? Because that C at that position will not base pair with the A in that position. You must have 100% base pairing. That's why the probes are very small. To hybridize, you have to have 100% base pairing here. So the normal probe sees the normal allele, but not the mutant allele. And then if we take the mutant allele and the mutant probe and give it to the mutant allele, watch what happens by PowerPoint. There it goes, there it goes. Oh, there it is. Notice how we get 100% base pairing of the mutant probe with that T, base pairs with that A, and all the other nucleotides base pair with each other. So the mutant probe sees the mutant allele, but not the normal allele. You must have 100% base pairing. Now what I'm going to do here is I'm going to outline, suppose a family with hemochromatosis, with a trait in their family comes into your clinic. And because we know what the mutation is causing hemochromatosis, suppose you want to analyze this particular family. You want to look at the gene directly in all the family members. So what's done, I'm going to outline the sequence of events. What's done is you take cheek cells from all family members. You just cheek cells, epithelial cells, put them in a test tube from all family members. And then you isolate the DNA, get rid of all the histones with detergents. And then you do PCR. Again, PCR is the basis of this thing called the dot blot. Just like the southern blot is the basis of the RFLP, the PCR is the basis of this type of technique called the dot blot. So you PCR amplify this region of the gene. Just PCR amplify that region. Just add primers and all the stuff we talked about earlier. So you PCR amplify the gene, and then what's done is to place that amplified DNA onto two spots on a piece of paper. Just take a piece of paper, a piece of paper right here, and I take a piece of paper, and so if it's my DNA, I spot it here, and I spot it there. 2 spots from each family member. And then after you spot from each family member in 2 spots, then you add what you do is the dot plot using the probes. All right, and then you can get results that look like this. Okay, now this is interpretation of the results. This is a dot plot, no electrophoresis here. The interpretation of a dot plot is very simple stuff here. The interpretation of the dot plot. Okay, now the idea here, the concept here is, in the interpretation of it is, by convention, if you see an open circle, that's telling you when you add the probe, nothing hybridizes. Nothing is being seen. If, on the other hand, you see a solid circle, either a black circle or a gray circle, it's telling you when the probe is added, it sees something. It's hybridizing with something in that spot. Okay, you must know this, okay, that what the circles represent. Open circle, probe sees nothing. Closed circle, the probe sees something. It's hybridizing with something. All right. Now the idea here is, if we take person number 1, and we have that person's DNA in 2 spots. In 1 spot, we add the normal probe. A solid circle is seen. If we add the mutant probe to the other spot, nothing is seen. What is your interpretation of person number 1? What's interpretation of person number 1? Not hard. That's right. He is homozygous normal. Look how simple that was. Simple. Now, if we take person number 2, in 1 spot from person number 2, we add the normal probe. Something is seen. Gray spot, solid circle. In the other spot, we add the mutant probe. Something is also seen. The gray spot. What's the interpretation of person number 2? That's right. He is a carrier. He is heterozygous carrier. Now, why gray versus black? It's the relative amounts. On board exams, they don't have to show you gray. If this is person number 2 who is a carrier, you might see a solid black circle here and a solid black circle there. Both spots lighting up or being recognized by the normal probe as well as the mutant probe. And then we take now person number 3. In 1 spot, we add the normal probe. Nothing is recognized. In the other spot, from person number 3, we add the mutant probe. Something is seen, the solid circle. What is the conclusion of person number 3? That's right. He is homozygous with the disease. Look how powerful that was. Look how powerful. There was no invasion of, no biopsy, no difficult thing you had to do. All you do is take cheek cells. I could take your cheek cells right now and PCR amplify that gene and then spot it on 2 spots and add these probes. And I could find out exactly what your status is for hemochromatosis, how powerful the dot plot is. Now, in your profession, the dot plot is only being used for a few diseases thus far. And the reason is you got to know what the mutation is that you're looking for. You have to know precisely what the mutation is. Why? Because the probes are specific for that mutation. So that's what. So in your profession, only maybe a half a dozen diseases. You could do this for sickle cell as well, but only a few diseases this is being used. But maybe 10 years from now, 20 years from now, you may be doing dot plots in your own office. How powerful the technique is. So again, you must understand the dot plot. And again, fortunately, in the assessment exam in genetics, there are several questions that show dot plots, and you will see it's not hard. How you interpret the dot plot is very simple. It's not hard. And you'll get it right as long as you don't make excuses. Understanding how you interpret the dot plot here. Sayed, you're too tired for the dot plot. What do you want me to do, Sayed? Stop in 3 hours and cut out all this material? I mean, You think you're tired? What about me? I'm here. I want to help you out. You can't make excuses. I'm too tired. You can't make excuses. You got it. You know you're gonna be a doc. Maybe you are a doctor. Or other people are doctors. You're gonna be in the clinics for hours and hours and hours, and some patient is under your care, and you're gonna say, "Hey, I'm too tired to work on you. I'm gonna go to bed." You're not gonna do that. You can't make excuses. You gotta man up and do it. You gotta do it. I'm after 4 hours. I'm still excited to be here. I don't know about you. I'm excited, but you gotta do it. Don't make excuses. The national board does not want to hear excuses. If I were to close after 3 hours, or because you're too tired, I'll be eliminating things, and then you're going to be heard on the boards. All right. Now, since it's 4 hours are done, we're done for the day. There are a few more figures that we'll cover tomorrow in genetics. So we're done for the day. We reached the 10 o'clock hour Eastern time. So we're done for the day. As the schedule says, in 4 hours we're done at the top of the hour now. And so tomorrow we'll come in, we'll do a few more figures in chapter 6. And then after that, we'll go back to metabolism, and we'll get into chapter 8 in metabolism and cover amino acids and that kind of stuff tomorrow. Okay, everybody. So please, I'm going to post several questions on the Facebook. Look at those questions, and then type. You don't make excuses. Go through those, and it's to your benefit. And then tomorrow afternoon, I'll post the answers, and we'll go from there. So anyway, everybody, hopefully this was beneficial tonight. I know we covered a lot of ground. I know, but you saw the high yield stuff. It could have been a lot worse, covering low yield stuff. But in any case, covered what you needed to know. Okay, everybody, you take care of yourself, and I'll see you tomorrow at the normal time. Take care. Have a great evening.